Hello, everyone, and welcome to the second day of the meeting. I hope the room will fill up and that the talks, I'm sure, will be even better than those of yesterday. So we are now in the session, Pathways and Crossroads to Healthy Longevity, the Promises and Challenges of Neuroscience, and I'm very honored to invite my co-chair and the first speaker of this session, Myriam Grunewald from the Hebrew University, to tell us about how undoing loss of microvascular homeostasis promotes healthy aging.
It works. Okay, so good morning, everyone. And I'd like to start by thanking the organizers, and in particular, Ilya, for his hard work and his dedication to put this conference on, and also for the opportunity to share with you what I'm doing as a vascular biologist about aging. My work, in fact, stems from a very old adage that says that a man is old as his arteries, from the 17th century. This Thomas Sydenham, just by hearing the heart of his patients and measuring their pulse, he understood that biological age is not the same as the chronological age, and that healthy arteries contribute a lot to the usefulness and the longevity, regardless of the chronological age. And this is not surprising for vascular biologists, but I think you don't have to be a vascular biologist to appreciate how this huge organ, by the way, this is a cast of the vasculature of a young adult, this huge vasculature supports literally all the functions, the other organs. And not only because these are tubes that provide all the blood-borne substances, including hormones, to ensure the cohesion with the organ functions, but also because, as we heard yesterday from Uri Alon, these are the highways that are used for the immune system during inflammation or during immune surveillance, and the point of entry is the capillaries to enter into these organs. And also because we and others have shown that the endothelial cells that are lining the interior of the capillaries, they are producing what we call angiocrine factors, which are secreted proteins that are important for maintaining stem cells, for example, or are involved in the process of regeneration of parenchymal cells. And what is aging if not losing these two properties, maintaining the regeneration potential? So it seems that the vascular aging appears poised to play a critical role in the overall physiological aging of the body. And maybe if we find a way to maintain these vascular functions, we could delay age-associated diseases. So how the vasculature is aging, I'm sure you all familiar with what happens to your large blood vessels, the arteries with the atherosclerosis, aneurysm, the loss of elasticity. All this is recognized as a factor for age-related disease and can be corrected by medication or lifestyle. Less is known about what happens to the microvasculature, which are these little tiny capillaries that are the ones that are in contact with our cells in the body, in the different organs. And one of the most prominent phenotypes that we see in aged tissues is the decrease of the microvascular density that in mice we can easily visualize by just injecting a fluorescent antibody that recognizes specific proteins in the lumen of the capillaries. And as you can see here in aged mice, you can see a decrease in the microvascular density. And by the way, it is also true for humans. Because if you look, for example, at the dermis of the skin of a young adult, 20 years old, you'll see a lot of very nice capillary network, which is reduced in the skin of older persons. And it's true for almost all of our organs in the body. So the idea that we had is to find a way to maintain this capillary density. And one of the proteins that is hypoxic regulated and is very important, that's why Professor Elekeshet, my postdoc mentor, name it the guardian of the vascular homeostasis, because VEGF is involved in all aspects of angiogenesis, of the formation of new blood vessels. It is also indispensable to maintain some organ-specific phenotype of the endothelial cells, and if we block the VEGF signaling in the heart, for example, of a mice, you can see here this is the capillaries visualized by immunostaining. They will decrease if we block VEGF after a while, and the restoration of perfusion will be done only if we release this blockade. So what means that there is no backup to VEGF. So indeed, we checked the VEGF signaling in aged mice, and it's also true for human tissues, and we could see that the phosphorylation of the main receptor of this signaling pathway is reduced in aged tissue, as you can see here. And so we devised a system, a transgenic system, where we can increase or maintain, this is the right word, to maintain a right level of VEGF signaling. I don't bother with the details. And we were able to increase a little bit more VEGF in the liver of these mice, which is secreted then in the circulation, where we can measure it and can impact the vasculature and other cells in other organs. And here I would like just to bring your attention to two things. One, the dosage. The dosage of VEGF is very important. We don't want to shake the system by huge amount of VEGF. We do that twice the amount of physiological levels. And the second is that we treat the mice, the system is on from 8 months and onward. So all what I will describe to you is about prevention. Yesterday we heard someone asking about what can we do with young people so that they will age, where they will have a delay in their aging. And we are now working also on initiating this treatment when the disease are already there to see if VEGF can reverse some of the phenotype. So the system was enough to maintain a young foal-like level of VEGF signaling, and that was enough to maintain the microvascular density. As you can see in the liver, which is okay because the VEGF is emanating from the hepatocyte, but also in remote organs like in the muscle, in the adipose tissue, in the bone marrow, and any organs we looked at. So this work has been published 3 years ago, and I will just rapidly summarize what we have found. And it was extraordinary to see that just by elevating a little bit VEGF in these mice was enough to delay most of the age-associated diseases that we could measure in our lab or by way of collaboration, and I will just rapidly go over it. So, for example, VEGF maintains a young-like body composition and fat distribution, and it's important for the abdominal adipose tissue, which is very inflammatory, but also the adipose tissue in the skin, which is reduced and cause wrinkles and lowered elasticity and everything. So we could see that in VEGF-treated mice, we could maintain a good body composition from lean to fat, and the distribution of the fat was maintained as in young mice. We could also show by placing our mice in metabolic cages that the VEGF-treated mice maintain a young-like metabolism, and if you need more details, I can do that later. And looking at the liver, we could see that the accumulation of fat that happens with age in the liver of mice and human was significantly reduced in VEGF-treated mice, as well as the enzyme that you can measure as a measure to liver injury that were really decreased in VEGF-treated mice. We also saw less senescence in the liver that we heard a lot about yesterday by Valerie. We look at the muscle of these mice, and by different assays, we could show also that there is an increase in the muscle capacity, in the muscle histology, and particularly mitochondria were improved in their number and in their quality. And finally, we looked at osteoporosis, and we saw that both in male and females, the bone mass were really maintained in VEGF-treated mice. And maybe because of a combination of good muscles and good bones, we could also see that the kyphosis of these mice is improved. Kyphosis that we all know when we look at our grandparents. And finally, just because of the time, all these phenotypes culminates in a longer lifespan, which is, in my eyes, not so interesting, but more evident and more interesting was the compression of the morbidity time. And if you remember, yesterday we discussed that to say that if we just increase the lifespan without compressing the morbidity period, this period of bad health, we don't earn much. But I think that with the VEGF treatment, and it's more apparent in females, by the way, we have succeeded in compressing this morbidity, this period where you get all these age-associated diseases. So just to continue, just a word about VEGF and cancer, because when we say VEGF, immediately in the brain of the people, you associate it with cancer, because it's true. Cancer cells produce a lot of VEGF that they need to recruit new blood vessels in order for the tumor to grow and thrive. So we were quite skeptic, because you need much higher level of VEGF to induce to provide the necessary blood vessels for the tumor. But we followed by MRI mice to detect the spontaneous tumors. And as you can see here, in a 24-month-old mouse, in this strain, we have a lot of tumors. They were delayed, or at least not increased in VEGF, in 24-month-old VEGF gain of function mice. And by meticulously look at all the mice that we use for the lifespan study, we could see that VEGF treatment not only not induce spontaneous tumor, increase the rate of spontaneous tumors, but it decreases it. Okay, as you can see here, both in males and females. So now, for the last 3 years, we have looked at mechanism of why this tumor incidence is lowered in these mice. And Priya, together with Jenny Wolinski, who is in the audience, has looked at the immune system. And as I will just summarize this. She was supposed to come with her poster, but she's not here today because she's not feeling well. And what she saw is that first the thymus, which is so important for T cells, is maintained, and which is involuting physiologically in old mice and in aging human. The functional mass of the thymus is preserved in VEGF-treated mice, even at the age of 27 months old, which is very old mice. And together with Yechezkiel Eliyahu from Alon Monsenego's lab in Ben-Gurion University, we could see that the VEGF treatment maintains a healthy thymic output. She could analyze CD4, CD8, recent thymic emigrants, and naive CD4 cells, and she could see that in most of the mice treated by VEGF, we have an increase in the number of these population in the blood, but also in the periphery, which is a good thing, because generally approach that maintain the thymus are not able to maintain the immunity, a good immunity in the periphery. And here we could see that in the spleen or in the lymph node, we have the same increase in naive T cells and recent thymic emigrants. Of course, she looked at the function of these cells, and they are all improved in VEGF-treated mice. And in particular, we could look at the exhausted T cells that are accumulating with age and that are expressing the PD-1 protein, which impairs T cell function. And as you can see here, we have both in the blood, in the lymph node, in the spleen, a decrease in cytotoxic CD8 T cells and CD4 T cells, particularly in the spleen, that do not express this PD-1, so that we'll be able to attack and eliminate tumor cells that express this PD-L1 and the senescent cell that, if you remember, Valerie showed that they also express this PD-L1 ligand. So that can explain why in VEGF mice we see less senescent cells. So I will stop about this VEGF, that counteracting VEGF is promoting healthy aging in mice, and hopefully with the help of Merck, we are producing an NAV, which is FDA approved, to induce VEGF specifically in the liver of these mice, in a way to translate these results to something that we could do in human in the future. So, but I would like to take, I think I have 2 or 3 minutes, to introduce a new model that I think is important in the aging research, which are organoids. I don't know if you are familiar with what is an organoid, but it's a new technology that has been developed in the lab of Hans Clevers in Holland. And it allows us to take a small biopsy of a human tissue and to process it and to grow it in a special condition in 3 dimensions that allow them to self-organize and to maintain the stem cells of the tissue, so that it enables us to grow this tissue, and here the stem cells are stained in green, to grow this tissue indefinitely and to break it and to expand it and to freeze it and to thaw it and to use it as a regular 2-dimensional tissue, but 2-dimensional culture, but now you have a 3-dimensional that maintain the cell-cell interaction and the cell interaction with the extracellular matrix that we heard a lot yesterday about its importance. So having that in mind, people are increasingly using these organoids for many basic and translational applications that are listed here. Most importantly, the FDA has approved this model as a valid preclinical model for drug testing, for example. But for aging, these models are not yet very much used. And one of the problems, I think, is the access to tissues, to human tissues that necessitate Helsinki and etc. So in the lab, we have a lab in Hadassah Hospital where we got all the necessary issuing to use these human tissues, and we are testing these organoids to validate them as a good model for preclinical for your anti-aging drugs, or just to, if you want to test a pathway, an intrinsic pathway in a specific tissue. And for example, what is interesting is that these organoids maintain their biological age. If you do an organoid from a young patient or an old patient, they will maintain their phenotype in culture, which is huge. And in collaboration with Dudit Tzfati from the Hebrew University, we have measured, for example, the telomere length of that in the cells that are grown as organoids in vitro. And we could see that an old organoid has shorter telomeres and a young organoid has longer telomeres. And when you divide these organoids, the telomere will shorten more rapidly than in a human tissue in the body, but it could really mimic the aging of the tissue. In collaboration with Centarix, a company that you will hear, I think, today or tomorrow, we are also exploring the transcriptome differences between these organoids generated from young and old patients. And we already know that these organoids maintain their epigenetic age, which is really nice. And I think that this a model can really be used to test anti-aging drugs or pathways that we think are important for the aging science. And we can think also in the future to use these organoids for regenerative medicine. It's not science fiction. It is already used in Japan to repair the intestine of people having refractory ulcers, for example. Experiments are done in mice by the time to correct the retina of people suffering from retinal disease. And we in the lab are doing endometrial organoids to try to correct the endometrium of diseased women. So I think that, I hope that I convince you that organoids are the next model, relevant, most relevant for the human to study human aging. And you are invited to contact us if you want to use the organoid that we produce. So to wrap up this lecture, I would like to thank all the people from the Eli Keshet Lab that have helped in this study. And I hope I convince you that we can correct the adage, a man is old as his artery, from the 17th century, to a man and a woman are old as their arteries and capillaries for healthy aging. Our collaborators and our fundings, in particular Merck and the Israeli Ministry of Science and Technology, that allows us to translate our results. Thank you.
Any questions?
Regarding VGF, microphone, please. Yeah. If I look in general for your presentation in VGF, we basically improve function by correcting for the levels of a protein that circulates. So if we look at the presentation at VGF, the main theme was improving function in relation to correcting a circulating protein. A while ago, it was suspected that growth hormone had those effects. So my question is, what do you think are other similar candidates that could be corrected to improve health span?
As a growth, kind of growth factors.
Yeah, I mean.
So for the vasculature, you have other growth factors that can do the job, but again, VEGF is indispensable because it is involved in all the aspects of neovascularization and also the maintenance of the organ-specific phenotypic structures that you see in endothelial cells, for example, fenestration in the liver or in the pancreas that allows free diffusion of hormones or lipids or things like that. So this fenestration. And what you say that it's not only correcting the circulating level of VEGF, but the VEGF also in the liver and in the signaling pathways of VEGF. Yeah. In the organs. We see that we have more phosphorylation of the VEGF receptor, probably in endothelial cells, but it's good that you ask this question because VEGF was considered as a vascular endothelial growth factor, but more and more we can see that he has receptors in many other cells, including fibroblasts, which are so important for secreting the extracellular matrix in every connective tissue. Or in immune cells, we have VEGF receptors also. So it's larger. We have begun with only the microvascular density that VEGF can correct or maintain, but we anticipate that the role of VEGF will be beyond all this microvascular density.
It has systemic effects.
It has systemic effects, yeah. Yes. Absolutely, yeah. And the dosage is important. Too much VEGF is not healthy, but the specific dosage is excellent for healthy aging.
Thank you. Okay, so thank you very much for Myriam for a wonderful talk. Our next talk will be by Zoom by Dr. Maria Cavinato from the Innsbruck University, and she'll tell us about senescence, skin and longevity, molecular mechanisms, and therapeutic avenues.
So first of all, I would like to thank the organizers for inviting me to share the latest results of my lab with you. I was really hoping to be there in person, but unfortunately this was not possible. But I hope that I can share with you today a little bit about cellular senescence, skin and longevity, the strategies that we are using to study mechanisms of skin aging, and also to address these senescent cells in the skin. So I like to start my talks by advocating for the study of skin aging. The skin is our outermost organ of the body, and it is the organ that is in contact with the environment. So it's a protective organ. And during aging, the barrier is decreased, which means that we are more prone to get diseases and damages from the environment. Also, skin is a social organ, and skin aging can also have a negative impact on self-esteem, body image, and therefore in quality of life. So therefore, it is very important to study skin age. I think that at this point, you have heard enough that there are several hallmarks of aging, and among them, the accumulation and persistence of senescent cells in the tissues is one of these hallmarks. But we are not just interested in cellular senescence, but we are also interested in another hallmark of aging, which is mitochondrial dysfunction. And we still don't know, and there is a lot of debate in the field about if mitochondrial dysfunction is a cause or consequence of cellular senescence, but they are indeed interconnected. And it is also known that accumulation of senescent cells in the tissues is leading to age-related diseases, and that xenotherapeutics that are targeting senescent cells either by killing these senescent cells or by inhibiting the activity of the senescent cells have some kind of rejuvenating power. We and others were already able to demonstrate that in the skin, during aging, there is an accumulation of senescent cells, both in the epidermis, which is the upper layer of the skin, as well as in the dermis, which is the lower layer of the skin. And we know nowadays, using organoids, that by the time that we see senescent cells in the dermis of the skin, we have an aging phenotype in the epidermis of the skin. And this is changing based on the percentage of senescent cells that we have in one compartment, which means that there is a communication between the cells of this tissue. And then I would like to go back a little bit about what are senescent cells. I think this was already quite explored in this meeting, but I would like to highlight that senescent cells don't have a single type of phenotype. They have several characteristics that are not common to all of the senescent cells, but most of them, they have an increased, they have altered morphology, they are growth inhibited, but especially for our studies, they secrete a set of chemokines, cytokines, matrix metalloproteases, and nowadays we know that also extracellular vesicles that are consistently together call it the senescence-associated secretory phenotype. And senescent cells also have, most of the times, impaired mitochondrial function. The issue of these senescent cells in the tissue is that when they are removed and they have this transient SASP, this SASP is beneficial for the tissue. However, upon persistent SASP and persistent mitochondrial damage that we know nowadays that is also inducing the SASP, these cells, they can be pro-inflammatory and cause chronic inflammation, tumor progression, and age-related diseases. So these are the two main topics that we are interested in our lab: impaired mitochondrial function and the secretome of senescent cells. Based on this, we have developed a strategy in which we try first to understand molecular mechanisms of cellular senescence and mitochondrial dysfunction. Based on this, we find targets for intervention, and then we are also trying to find strategies about how to intervene on the skin. So in the next slides, I will share with you two targets that we have found based on molecular mechanisms, and I will discuss with you the strategies that we are trying to develop to intervene on the skin. The first target was based on a model that we call the UVB-induced senescence of human dermal fibroblasts. This model In this model, we irradiate human dermal fibroblasts with UVB twice a day for 4 consecutive days. And at the end of 2 weeks, we have a full senescence phenotype that is characterized by growth arrest, increased activity of senescence-associated beta-gal, and expression of senescence-associated proteins. So based on this model, we have started studying mitochondria physiology, and we have seen that UVB induces fragmentation of the mitochondria. And this fragmentation of the mitochondria is initial step for the elimination of this fragmented mitochondria by mitophagy. We have then nailed down the mitophagy receptors that would be acting on this elimination of damaged mitochondria, and we could find out that the mitophagy receptor NIX was not just upregulated by UVB, but also we changed its subcellular localization. So in control cells, the NIX was mainly localized in the nucleus, while in irradiated cells, NIX migrated to the mitochondria and primed this mitochondria to be eliminated by mitophagy. So there we found a target. Based on a molecular mechanism, we found a target for intervention. So when we do this, what happens is that we knock it down. So we transduced cells with lentivirus to deplete NIX in our cells, and when we irradiated our cells, our NIX depleted cells with UVB, we could discover that indeed NIX is a essential mechanism for the development of senescence, and in the absence of canonical mitophagy, the majority of the cells were not able to cope with the UVB stress, and they were not going to senescence. Additionally, these cells started secreting extracellular vesicles, and these vesicles contained damaged mitochondria, suggesting that this mechanism is an alternative mechanism of quality control to complement the absence of the canonical mitophagy. But simply independent of UVB, knockdown of NIX, so depletion of this mitophagy receptor that is responsible for the elimination of damaged mitochondria upon UVB, already leads to cellular senescence, to premature senescence in human dermal fibroblasts. And this premature senescence is also characterized by accumulation of damaged mitochondria and also by secretion of extracellular vesicles containing damaged mitochondria. So based on this target of intervention, we have developed our organoids, as it was explained by Myriam before. So these are very important tools for the study of physiology of tissues. And in our organoids, we seed the fibroblasts in a collagen matrix. And then on top of this fibroblasts, we seed the keratinocytes. And after these, we put these organoids in what we call air-liquid interface. This is ALI, and this means that the epidermis will be in contact with the air, while the dermis will be in contact with the medium, mimicking the conditions of the skin. And after 7 days, we have a fully developed skin model. So we have prepared, based on these mechanisms of intercellular communication, we have prepared organoids containing always wild-type keratinocytes and either wild-type fibroblasts or NIX knockdown fibroblasts. And before air-liquid interface, they looked more or less the same, but 24 hours after putting these organoids into air-liquid interface, we could already see macroscopic differences in these tissues. And when we analyzed the histology of this organ, we could see that the organoid prepared with wild-type fibroblasts developed a completely normal epidermis, while the ones prepared with NIX knockdown fibroblasts developed epidermal layer that is very similar to squamous cell carcinoma. And we have to think that this was caused by cells that were in a different compartment. So most probably the mitochondrial defect that is happening in these cells that triggers this release of EVs containing mitochondria influences somehow in the phenotype of the skin. So this is one of the targets that we found so far. and that I would like to share with you. The second target comes from a different model. It's a model of TBHP-induced senescence. So TBHP causes oxidative stress, and we have developed this model in which we treat our cells with TBHP, similar to the UVB, twice a day for 4 days, and at the end of 14 days, we have a full senescence phenotype. And this senescence phenotype is a little bit different from the UVB-induced senescence phenotype. And by doing transcriptomic analysis, we have found that a gene that is called GDF15 was highly upregulated upon senescence induced by TBHP. GDF15, I think you might have heard about this. It's considered a cytokine, and it's considered also a novel systemic biomarker of oxidative stress, inflammation, and cellular aging. And it can have beneficial and detrimental effects in the tissue, depending on where it is acting. So since this was a target that we found regulated in our model, we decided to modulate this target. So again, we generated these GDF15 knockdown cells. And here we also found that knockdown of GDF15 induces premature senescence. But interestingly, this premature senescence of the GDF15 knockdown is characterized by a different type of mitochondrial dysfunction. In this case, our mitochondria, they are in the GDF15 knockdown cells, they are hyperfused. And we know that the fission of the mitochondria, the fragmentation of the mitochondria, it's one of the first steps for these damaged mitochondria to be eliminated by mitophagy. And in this case, we have hyperfused mitochondria, but we also have a different secretome of these cells that is characterized by upregulation of several matrix metalloproteases, but also by the secretion of extracellular particles. And here I say particles because in contrast with the NIX knockdown cells, GDF15 knockdown cells that cannot do fission on their mitochondria, they mainly secrete cell-free mitochondria. They secrete several of these cell-free mitochondria in the extracellular environment. And what happens when we make these organoids with GDF15 knockdown fibroblasts is that the secretome of these cells modulate the skin tissue towards an aging phenotype, which means that they have decreased epidermal thickness, they have more apoptosis of the epidermal cells. And again, this is triggered by cells that are in a different compartment. So these are two different molecular mechanisms that we have found. And based on these, we found targets for intervention in the skin. And we are now trying to modulate these targets. And how do we do this? We do this together with the Institute of Pharmacy and Pharmaceutical Chemistry of our university, together with Dr. Teresa Casares. And when we find our targets, she goes back to the chemical space and she finds compounds that are already existing and are able to modulate the pathway that we are interested upon. And she works on compound optimization based on the properties of the existing compounds and the specificity of the pathway that we want to investigate. So far, we have 3 compounds that we are using for modulating GDF15 activity in the cells. And as you can see, compound 2 has a very promising activity. So it kills more than 60% of senescent cells and around 10% of control cells. And now I would like to actually tell you a little bit about issues that we have, because it's very interesting to find targets for intervention and compounds that we can use for intervening in the skin tissue. But we have 3 problems that always come to our hands when we are talking about interventions on senescent cells. The first problem is how to detect senescent cells in the skin, because so far, the main way of detecting senescent cells in the skin tissue is via biopsies. But if we think that we have to take biopsies from the elderly or people that have problems in wound healing, this is not really the best way to assess senescent cells. The second problem is that we know nowadays that senescence is not a homogeneous phenotype. So even within the same tissue, within the same cell types, senescence is very heterogeneous, and we have to be able to find biomarkers for assessing or mechanisms that we can really assess senescent cells. And third is that, as I told you at the beginning, skin is the barrier of the body. So normally, the compounds should not enter the skin. So we have to think about how to effectively reach the target, like the skin cells. So to circumvent the first and the second problems, we are working together with the Leibniz Institute in Jena for the development of non-invasive approaches to detect senescent cells in the skin. So we are using a set of imaging, different kinds of imaging, together with machine learning, such as Raman spectroscopy and microscopy, near-infrared spectroscopy, and optical photothermal infrared microscopy, among others. And so far, we are able, in 2D cultures, to completely differentiate with non-invasive approaches, completely differentiate between control and senescent cells, and also using optical photothermal infrared microscopy, we are able to differentiate between composition of proteins, lipids, and DNA between senescent and non-senescent cells. And when we are using our 3D skin equivalents, we are making them with different proportions of senescent cells, because we have to think that in the old skin, we don't have 100% of senescent cells, and the system is already able to detect and to differentiate between skin equivalents or organoids that are prepared with different proportions of senescent cells. So this is something that we are investing a lot of energy. And the other issue that I told you is how to intervene, because we have to guarantee that these compounds or this way, the modulation of the system has to reach the cells that we are interested in. In our case, it's mainly the fibroblasts. We are in the dermis. So we are also investing a lot of energy on developing new approaches for skin intervention. And we are now researching therapeutic nuclear magnetic resonance that was already shown by colleagues of ours to modulate mitochondrial physiology in cells. So this would be a non-invasive methodology of manipulating mitochondrial physiology in our cells and trying to recover their function. I hope with this I could convince you about the importance of the study of cellular senescence and skin aging, to show you a little bit of our strategies to address these issues, and the efforts that we are making to find strategies for addressing senescent cells in the skin in non-invasive methodologies, and also how to target our senescent cells. And I would also like to thank my group that is very supportive in this work, and all the people that are helping directly or indirectly in this work. And thank you for your attention.
Thank you very much. Do we have any questions?
Maybe I will ask. This wonderful talk. And do you think that you can detect senescent cells, extracellular vesicles derived from skin senescent cells as a liquid biopsy in the blood of the
So in the blood, it's very difficult, because we have to make sure about the origin of these extracellular vesicles. And so far, there is no unique biomarker to define the origin of extracellular vesicles coming from the skin. But this is a new field, and I'm sure that we and others are putting a lot of effort on developing this technology. But what we can is, so far, to isolate extracellular vesicles from the interstitial fluid of the skin. It is a very painful and long process, but it is possible.
Thank you.
I also have a question. I was wondering, what do you think happens to these extracellular vesicles carrying the mitochondria, are they delivered to other cells? Do you think they are toxic to the body?
Well, we have preliminary data that shows that they are uptaken by healthy cells. And we are now trying to investigate if they fuse with the mitochondrial network of the healthy cells, or if they just release like a hermetic response. So this is ongoing research.
Okay, thank you very much.
Okay. Thank you very much. And I think we should, yeah. Would you like to open your? Yeah. So I'm pleased to introduce Sivan Henis-Korenblit from the Bar-Ilan University, who is going to tell us about using model organoids to discover genetic strategies towards improving health spans in humans.
Okay, so thank you so much for coming and for the organizers for the invitation. And I'm happy to tell you about work that we've been conducting in my lab over the last decade using the model organism C. elegans to discover new strategies towards improving health span using genetic approaches. So as you all know, and as we have heard yesterday, as modern medicine progresses, we live longer and longer. And as we have managed very well to extend our lifespan, we still suffer from many diseases at old age. And we even suffer from them more than we used to, because now more and more people reach this old age. And one of the hallmarks of many of these age-related diseases is the accumulation of misfolded proteins, and which is very, very harmful, especially to our brain. So we suffer from many neurodegenerative diseases in the last decade of our lifespan. Interestingly, these proteins that form these aggregates and cause disease, they're not proteins that are expressed only in the old, they're expressed throughout our life, also when we are young. But when we are young, these proteins are not harmful, they do not form aggregates, and they contribute to our physiology. So it's not that these proteins have to cause disease, and when we are young, we can deal with them and prevent them from gaining a form which causes disease. But when we are old, we lose this protection, we lose this ability, and then we suffer from the consequences of disease. So while it's a very important aim to extend lifespan, we also want to extend health span and especially maintain health span in the new old age that we now reach. So in my lab, we use the model organism C. elegans to study aging. It's a great model organism to study aging because it ages relatively fast. It has a lifespan of about 2 to 3 weeks, and it ages during this lifespan. So we see all kinds of age-dependent physiological collapses that occur in this organism. For example, defects in the reproductive system as we age, loss of motility, and at the end, death. But one of the first age-dependent physiological declines that we observe in this animal is a collapse of proteostasis, the collapse of the ability to maintain proper folding of proteins. So we see age-dependent sensitivity to proteostasis stresses like heat and toxins that interfere with protein folding. We see age-dependent misfolding of metastable proteins, protein that are on the verge of folding correctly. They can fold correctly with help or fold incorrectly. As we age, more and more, a bigger portion of them folds incorrectly. And consequently, we see age-dependent protein aggregation, which is the aggregation of those misfolded proteins. So this event of proteostasis collapse is very conserved in C. elegans. It happens relatively early, right at the peak of reproductive maturity. And it's a very, very blunt decline in physiological function. It's not something gradual, but rather one day they are fertile adults and the ability to properly maintain protein folding collapses all at once. So we can use this system to study this phenomena of age-dependent proteostasis collapse. So what we are interested in my lab is to understand the process of age-dependent proteostasis collapse and find ways to overcome it or to prevent it. And one of the approaches that we take is the approach of unbiased genetic screens, which has the advantage that of finding new things and new pathways that no one has considered before. So over the last decade, we have performed several unbiased genetic screens in C. elegans to identify mutations in genes that can improve protein folding in general and improve protein folding specifically in old animals. So one of the screens that we have done was a tunicamycin resistant screen. Tunicamycin is a drug which induces protein folding defects in secreted proteins. And if we give the worms enough of this drug, they fail to develop. If we introduce mutations into the worms, we can identify mutations that help the animals deal with this toxin and isolate grown-ups and characterize them. This is a developmental assay. Like I said before, the ability to deal with protein folding is very different in young animals and in old animals, and therefore it was important for us to do screens for enhancing proteostasis, not only in young animals during development, but also in old animals. So we looked for markers that show us signs of proteostasis collapse visually. For example, the ability to express protective chaperones. If we are unable to express them in the old, this will likely lead to protein misfolding. So we would like to find mutations that help express chaperones in the old. Or we can identify secretion defects in the old, so proteins that needs to be secreted remain trapped in the producing cells. If we can find mutations that help these proteins be released from the secretory system into outside of the cells, then that would be a sign of recovered proteostasis. So we have all these visual markers in animals. We let the animals age after exposing them to all kinds of mutagenesis treatments, and we identify the mutations that help restore proteostasis in the old. So I want to give you two examples of things that we have found using this approach. One of them will be from the developmental screen, and one of them will be from the older age screen. So in the developmental screen, when we challenge the animals with really high levels of proteostasis stress, we managed to isolate very, very few, about 20 different strains that managed to deal with the extreme high stress that we exposed the animals to. And we sent these mutants for sequencing and identified the mutations and confirmed that the mutation that we identified were associated with the enhanced proteostasis resistance. And what we found was that many of the mutants that we identified had mutations in similar genes. And even if some of the genes are not exactly the same genes, they all serve in the same function. These are all genes that are required for the proper function of a sensory organ in C. elegans. They build a cilia through which the animal can sample volatile and gustatory signals, food and smell, and by that the animal recognizes the environment. So all the mutations that rendered our animals super resistant to the stress were mutations that basically interfered with the ability of the animal to smell and to taste, kind of like after you have bad corona. So these animals that lose all sensation, not only they don't smell and they don't taste, they also don't see and they don't hear because they are worms that live in the ground, so they don't have these organs. So basically these are animals that lose most of their senses and remain only with the sense of touch. And strikingly, what we found is that while normal animals fail completely to survive under the stress conditions that we expose the animals to, animals that have this sensation problem were completely resistant, 100% resistant, as if they were not exposed to any stress at all. We thought at the beginning maybe they're not getting the chemical that is stressing them, but we confirmed that they do activate stress responses. So they definitely are exposed to the stress, they respond to the stress, but they're very, very, very resilient to the stress. We did some molecular analysis, and we found that this super resistance, which is really some amount of resistance that we've never seen before, is a result of a combination of several protective pathways acting together and synergizing. Some of these pathways include the UPR, dedicated pathways aimed to deal with misfolded proteins in the secretory organ of the cell. Another pathway that contributed to this resistance is the FOXO-DAF16 transcription factor. It's a stress-related transcription factor that its activation promotes stress resistance and longevity, and there was a strong reliance on autophagy. And you needed all three components to get this super strong resistance. So after being excited that we found this really resistant manipulation that we can do to the animals and get proteostasis resistance, we wanted to know, is this really good for the animal? What about the health span? So we went to explore health span criteria in the animals which harbor these mutations. We started by looking at muscle tissue, and surprisingly, it seemed that the muscle tissue is deteriorating when we interfere with the sensory organ rather than getting better. You can see here muscle fibers in normal animals which have a nice organized structure, but if we introduce mutations that interfere with the sensation of the animals, we suddenly see this collapse in the organized structure of the muscles. If we look at a polyQ model, which represents protein aggregation, which increases with age, we can see that animals which has the sensory defect have more protein aggregation, more foci than normal animals. So we are very surprised by this result because we're expecting animals that can deal with stress should have better physiology. And we kept looking at more and more proteostasis models in the muscle, and they all showed this persistent bad result, and that is that not only is the muscle physiology not improving, but it's even getting worse. At some point, we looked at other tissues outside of the muscle. So one of the tissues that we focused on was the intestine. And for our surprise, here we saw the complete opposite effect. If we look at the polyQ model in aging worms, we can see that normally they get these polyQ aggregates in their intestine, but if we introduce now the sensory mutation, much less of the animals have these aggregations, and they seem to be doing better. We also can do a test of the integrity of the intestine, and you can see that with age and upon exposure to stress, normal animals lose the integrity of their intestine, and you can see all this leakage of the blue food out of the intestine, but animals with the sensory cilia defect, their intestine remains very, very tight and functional. So what we learned from this screen is that it is, first of all, that it's possible to achieve extraordinary resistance to proteostatic stresses, but we need to bear in mind that proteostasis mechanisms differ between different tissues, and what is good for one cell type or one tissue may not be good and even harmful to another tissue. We think that this model demonstrates that proteostasis can be regulated across tissues. We did a manipulation in the neurons in the brain, in the sensory input of the animals, and the outcome is changes in proteostasis in other tissues, in the intestine and in the muscle. And we think that these results tell us that sensory information is important for choosing a stress strategy. We think that when the animals are have their senses, it's important to keep their muscles intact because then they can sense the environment and take insightful decisions where to go to run away from the stress. But if running away from the stress is not a possibility, not because you can't run, but because you can't choose, you don't know where is it better to go, then you have to stay put in place like a plant, and then you have to work on your resilience. And we think that the intestine might be the soft spot of this animal, and therefore it invests in intestinal resilience. So this was the main finding of this developmental screen that we have performed. Like I've said, we've also done other screens, specifically in adult animals. And I want to share with you a manipulation that we have identified that can improve proteostasis, specifically in old animals. And the mutation that we found was in a gene called PANC1 in C. elegans, or PANC1 in mammals, and it's not pink, it's not the mitophagy it's not related to mitophagy, it's related to the biosynthesis of a very important a cofactor in the cell, namely coenzyme A. So coenzyme A is a central molecule in the cell. It's a cofactor which participates in hundreds of biochemical reactions in the cells. It's very, very prominent in the mitochondria, and it's important for mitochondrial respiration. So we found that mutations that mildly disrupt PUNC1, that means that they should lead to mild deficiency in coenzyme CoA, are good for proteostasis. We did several functional assays in aging worms, and we found that in the poly-Q assays, inactivation of this gene prevents protein aggregation both in the muscle and in the intestine. It helps metastable proteins stay and maintain a better fold. It helps animals survive stresses, acute stresses like heat shock and tunicamycin, like I've shown you before. So unlike the previous talk, which had very specific effects on different tissues, here we have a global improvement of the ability of the animals to deal with proteostasis challenges as they age. We wondered if this is true only for worms, because this is a very important pathway, and having CoA is very important for so many functions. We wanted to be sure that it's not only limiting it is not only good for worms, but also for human cells. So we collaborated with the lab of Yaron Shavtal, and we exposed U2OS cells to heat shock. Exposing the cells to heat shocks induces apoptosis, but if we do so in the presence of an PUNC inhibitor, the cells are much more resilient to heat shock. So I think this phenomena is, first of all, evolutionary conserved, and second of all, it is a cell autonomous phenomena, because now we can improve proteostasis in isolated cells outside of the context of a whole organism. And we also delved into the mechanism of this improved proteostasis. And what we found was that improved proteostasis was not dependent on the proteasome system or the lysosome system, which are the protein degradation systems of the cell. So even if we don't have functional proteasome or lysosome, we still get proteostatic benefits when we inhibit the levels of CoA. But the improvement was highly dependent on the presence of specific chaperones. If we interfere with the induction of these chaperones, then we lose our benefits. And we can demonstrate that limiting CoA levels potentiates the increase in the levels of these chaperones. And we identified a transcription factor, HLH30T-TAB, which is important for this high synergistic levels of chaperones. So to summarize, I've shown you the efficiency of using genetic approaches to identify points in which we can manipulate proteostasis in the animals, and we can find all kinds of manipulations, mild manipulations that can improve proteostasis across tissues, or extraordinary resistance, which focus on specific tissues. Some of these mechanisms are regulated cell autonomously and some cell non-autonomously. Overall, our ability to identify mutations that improve proteostasis, I think, is very optimistic, and it means that we can find ways to resolve the proteostasis issues that we have at the end of our life, and in that way generate better health span that will go together and accompany the extended lifespan.
Are there any questions?
Okay, great talk, thank you. I know I'm not very big expert in this field of C. elegans aging, but I've seen a couple of papers by James and colleagues they demonstrated that age-related death of C. elegans depend on its kind of program. It depends on the certain change of functioning of its intestine. It's being digested by the worm to form more eggs, to form yolk. So from your first part of experiments, it seems like your mutations could interfere with this process. So you just break down the process of dying of these
So
animals.
some of our, C. elegans, the embryos of C. elegans, they get their yolk from the intestine of the animal, and that's a very imposing process on the animal. So we did some of the experiments in animals that have a mutation that removes away the reproductive system completely. So those animals don't have embryos, and therefore this load, the need to feed the animals does not exist in these animals, and we still see improvement in the proteostasis in these animals. So we think that's one aspect of the aging of this animal, but we have here improvements that are beyond that.
Hi, thank you for the talk. I worked with C. elegans for 3 years, that's close to my heart. So I had like one qualitative question, which is like, did the worms seem healthier? Right? I know you can do motility assays at the end of life and see if they're moving more, but also you can just look at the worms sometimes and see like, oh, these worms look pretty healthy compared to their counterparts. And the second question was, outside of like the proteostasis challenge, like, were they healthier? You know, like.
So actually, the two genes and the two manipulations that I discussed are different in that sense. So the first manipulation, which is the intervention which interferes with the sensory input, that was shown in the past by the Kenyan lab to extend lifespan greatly, and these animals are very, very healthy and functional. For the mutation in the PANC1, other effects, the proteostasis improvements there were consistent but mild. They were not accompanied by lifespan extension. So in that case, I think it's more of a health span improvement rather than a lifespan improvement.
Sivan, we have a question from someone from Aneta from Zoom. And she's asking, is attenuated metabolic process by mild disruption of CoA, is it good for resilience?
Is it good for?
Resilience.
Resilience. So it was good for resilience. The manipulations of the PANC1 pathway, every proteostasis assay that we looked at, they performed better, but it was always a mild increase. So it's not 100% resistance, 10% increase in the resistance. And we were limited for doing, increasing the manipulation, because at the end of the day, you do need a minimal amount of CoA to be healthy and functional.
Mm-hmm. Any other question? So I think we have a coffee break now. I think Ilya is not here. Thank you, Sivan.
Thank you.
Good morning. I would like to invite the first speaker of this session, Professor Gil Atzmon from Haifa University, to tell us about non-translational approaches to longevity models.
Thank you. I would like to thank the organizer for inviting me to speak to you today. By the way, it's the University of Haifa, not Haifa University. It's the only university it's to separate with us, between us. So I would like to convince you, one moment. Okay. I would like to show it to you today a non-traditional approaches or different model for longevity. And I would like to start with to acknowledge all the guys that support this study, particularly Mariana Andrus, a PhD student that just finished her PhD this year, and of course, Abraham Giller. צבי, רק תן לי 3 דקות לפני הסוף. So, the problem is that we invest too much time in order to find a model that suits for longevity, and the main problem with longevity, it's something that you can, it's more of a dream that you sell the person, because you interact with the people that are 30, 40 years old, and you're talking about a trait, a phenotype that will happen in let's say, 4 to 5 decades later. So the question, what is the best animal model, so-called, that we can try to study and to follow up and to understand better the biology behind it. Let's start with what we discussed within the 1.5 days since we start, and we're going to discuss it later in this conference. And this is animal model. Most of us use animal model in order to find a trait of interest, the study to understand the trait of interest. And we heard about, worms. We talked about, flies, dogs, mouse, and rats, and even monkeys, okay? However, the problem with this animal model is that they have, the pros are the short lifespan, which means we can point an MS student or PhD student or even postdoc, and it will capture the beginning and the end of the life of this creature. And with this, I'm talking about short lifespan, generation time. We can manipulate because this is animal and we can do whatever we want. And we can intervene with the lifestyle of this animal, again, because we can. The problem is they are not human. So the translation between what we study in this animal model, we will always need to take with care, with conscious. It's not all the time translatable. So let's start with human model. And here I'm talking about a disease of accelerate aging. The most known one is the progeria. As you can see, those two boys are around 12 to 13 years old, and they're already at the end of their life. This is accelerate aging. There are other syndromes like this, Widerman, and all those 4 that I just listed here. The problem with them is that this is a pathological state. So it's not healthy. So how can we translate what we find there in healthy aging? The generation time is okay because they live short. Manipulation, somewhat we can do, and some intervention we can do. However, remember, those are humans, so we are limited in the way we can do or we can interact with these people. But the good thing on the process, I call it, it's they are human. So the translation is easier. So let's go for the longevity, the guy who lived to 100 plus. So those are the right model to study healthy aging because they are healthy people and they present the trait that we look for. However, they have long lifetimes, so in order to capture what change, you live the same amount of years as they are. Generation time is very long. You cannot do manipulation and you cannot do intervention. However, they are human. So if we take only the concept of those 3 so-called animal model per se, it shows design, but it's not human model, and there either you can have the pathology or not. Accelerate aging, it's show design, it's human model, but they have the pathology, so you cannot translate it into healthy aging. And longevity, it's not a show design, but it's a human model and doesn't have pathology, meaning healthy aging. So, as I said, animal models accelerate aging and longevity all have advantage and disadvantage in terms of becoming a realistic, translatable, short-lived human model capable of replicating the aging process in a short period of time. To overcome this obstacle, disadvantage, we propose pregnancy as a translatable, short-term, and natural new human model for aging, which demonstrates several similarities in physiological and cellular degradation mechanism. What I'm saying about this, it's when women become pregnant, we talk about her physiology, not the fetus. Because every trimester, there is progression with the degradation, the physical degradation. Because all the energy, all of the physical physiology of the woman invested in the growth of the fetus. So this takes a toll on the female who carry this fetus. And here what we can do is to follow this degradation over time. And when you look to it, you can find a lot of similarity between pregnancy and aging. So, as I said, pregnancy, it's a short design, it's only 9 months. It's a healthy human model, and every woman will agree with me that it's a healthy state. It's not pathological, and as I said, it's healthy. Just to give you a core of similarities between pregnancy and aging, and there is a lot more, we're talking about inflammation, we're talking about accumulation of free radical damage, DNA and protein. We talked about pathological and telomere, which can be a biological marker for aging. And we publish it in aging research. We evaluated similar process between aging and pregnancy by comparing common biomarkers, pathologies, genetic, epigenetic effect to establish the progeny pregnant body as a model for aging. The link is made in terms of current view in aging as byproduct and natural selection, as well as unrelated molecular parallel between the two, relying on existing research and mode of human in other animal when applicable. And last one, we showed that the response of pregnant body to this change and through that illuminate unique areas of potential study to advance our knowledge of the maladies relating to aging and pregnancy and an avenue for solution. So this was a theory. The question, how we go from theory into practice. And this was the next slide. I will show you what we gain. In this scheme, we took 3 groups, centenarians, offspring, and the control. We used the Affymetrix platform in order to capture the CNVs within those people and to find if there is differences between centenarians, offspring, and control. Offspring is the offspring of the centenarian. Just to capture your interest, copy number variance, it's the same segment that goes within individual, and the change between them can tell you if there's something happened. We next, after we found counted CNV, we did the analysis, we scanned for CNV that distribute differently between centenarians and control, and we took those candidate CNV and went to mice model in collaboration with Chaim Lab. We did quantitative PCR to those guys, meaning the pregnant mice, and we measured the gene expression as well as the DNA copy number. Those were the CNV homologs in mouse model and the adjusted genes, meaning we not just look for the DNA marker, we look for the adjusted genes that are located next to them in order to see if these changes in CNVs affecting the expression of those genes. As you can see, כמה זמן יש לי? Okay, so we saw differences even number between centenarians and controlled blood sample. As you can see, there are 7 of those. They differentially changes. And if we pick on one of them, we can see that there is difference, differential CNV between case and control. Remember, control are 70 years old and centenarians are 100 years old. And we can see that there is a significant difference of CNVs in this loci between centenarians and control. We look at the gene expression and we saw that there is differential difference. And here I want to draw your attention into this scheme. What you see here, this is what we look for a trend. It's either it's go up with the trimester, and after labor it goes down, or the other way around, it goes down with the trimester and go up after delivery, and this counts not just for the CNV, it's also for the gene expression. And the idea is to capture the degradation of those markers with the age of the pregnancy. So what you can see, the CNV trend during pregnancy stage, you can see that within this loci, we see increase and then somewhat decline, and this is more vivid when you look at the expression. Okay? It goes down and goes up after delivery. We saw it in another 8 candidate CNVs, and of course with the genes that adjusted to those copy number variants, and you can see there is degradation and increase, degradation and increase, and here it's incline and then decline. So we get trend for both sides. And to finally, what we look for is the epigenetics through methylation. And as you know, the interaction, epigenetic is the interaction between the DNA and the environment, and it can be a result of cosmic effects, surrounding environment, body, organ, tissue, cell, and organelle. And the combination of the environment, the environmental signal and the DNA signal will give you the phenotype of interest. So if we look to it, what you can see, the similarity between aging and pregnancy. You can see that there is decline here. See the level of yellowish until the 3rd trimester, and there is increase after delivery. The same happened here between 50 to 60, and the yellow it's around 80 to 87, but at the age of 95, which means the longevity one, we see an increase in those expression. If you take one of the example, this CNV, you can see that the degradation and then decrease at the age of 95. And if we look at the pregnancy, we can see the same degradation and then increase. With this series of experiments, we propose pregnancy as an improved and more effective aging model that may help us comprehend and complicated feature of healthy lifetime. An additional outcome of this model is that since postpartum recovery age in completion of reversal mechanism, this mechanism may be applicable to aging treatment, delaying the aging phenotype with also reversing aging process. which means if we can translate the end that we saw, the increase of those signatures to aging process, and like in the pregnant lady, she almost recovered to 100%, we can do this effect on aging or partially on aging, and we can decrease the red zone, which is between healthy lifespan and lifespan. Thanks.
Did you by any chance differentiate between first pregnancy or second or third pregnancy? Or the age of the pregnant lady?
It's a good question. We just started, and we wanted to show that this hypothesis works. So when you want to show that the hypothesis works, you take all the green light. Then we can, use more modified models to see if it's working as well.
Thank you.
Thank you very much for a very challenging lecture. And I'm sorry, maybe the question is stupid, but the very straightforward idea. It's well known that pregnancy is associated with immune suppression. Yes, to preserve the fetus is kind of, because it's kind of alien object in the woman body. So can't be, and the same thing occurs with age. Immune system is suppressed. So maybe what you observe is only...
Absolutely, this is exactly what I show.
So
that with the immune system degradation within aging, it's almost parallel, almost similar to what happened in pregnancy.
Maybe that's what you're studying.
Yeah.
Because it's very complicated system, a lot of genes involved, a lot of transcription involved.
Absolutely, absolutely.
So it's kind of study of immune deficiency with the age.
You want to do it? I accept.
Hi, thank you for the talk. I always enjoy listening to your talks. I think they're very interesting. I had, guys, two technical questions. One was like, how do you overcome the fact that the data that you're taking from women is longitudinal, but the data that you're getting from, I assume, from the older people is cross-sectional, right? So you're not really following like a guy for 20 or 30 years to see where the genes go. And then the second question is also pregnant women are under intense metabolic stress, right? Like, I think compared to like ultra-marathoners in terms of like the amount of calories they burn each day. Is there some consideration there also to how that affects gene expression?
So I first answer the second question, which is stress. And I can guarantee you that everyone agreed that aging is stress. So it's not just stress for 9 months, it's stress for 40 years. And for the first one, as it depends how many person you have in this age group. If you have only one per year, it's a problem. But if you have thousands of people that show the same pattern, then it's much more accurate. And also, if you look at and in this case we did it, at a person we looked at them longitudinally for a couple of months, and we take another sample and we compare and we saw the same trend. Okay? איליה רוצה לדבר.
Thank you. It's an historian of aging research. I remember that back in the 1970s, Vladimir Dillman made some parallelism between pregnancy and aging, and he related it to disruption of homeostatic regulation, especially the function of the hypothalamus, kind of elevation of sensitivity threshold. Do you also relate it to this parallelism to central regulation, to the role of the hypothalamus, to sensitivity changes?
It's a good question, and this is exactly what we did in the paper. That I didn't elaborate on all the systems that we compare, but there is much more, and there is a link to our paper in ARR in 2020 that do this comparison with any system, almost any system that we look for, And I think the only difference, which means there was no degradation, it's in the urine system. I'm not really sure about it, but I think this is the only system that in pregnancy, it's increased rather to decrease compared to aging. But as you said, yeah, there is parallel there as well. No?
Let's thank the speaker.
So thank you, Gil. I want to introduce our next speaker, who is the Dr. Zvi Bar from the Faculty of Medicine in Tel Aviv University, and he will talk about epigenetic information. Epigenetic information loss is a common feature of multiple diseases and aging.
I'll actually try this one instead of that one. So I'd like to thank the organizers, especially Ilya, for generating such a great conference in these challenging times, and the funders. And forget about the long headline. Let's start with something really simple. So I was analyzing some methylation data, linking methylation profiles in about 500,000 sites with kidney filtration rates. And I sorted the data based on the correlation, and this was the top hit. Nothing unexpected. You have a specific site. It has a nice correlation with a filtration rate in the kidney. But then we found something unexpected. Looking at the distribution of methylation throughout tissues in the body, we saw that actually this site is methylated in almost all tissues, except kidney, where it has moderate levels of methylation. At which point, I didn't see many sites that look like this. I asked, is this a coincidence? Did I just happen to pick a site that looks like this, or is this a general trend? So we went to the Chinese National Epigenome database, and we analyzed, this is from thousands of individuals, 30 tissue types. We identified all samples, all methylation sites that look like this. And we found 253 sites that look like this, lower methylation in kidney and higher in all other tissues, and 36 sites that look the exact opposite, high in kidney and low everywhere else. And it turns out of these sites, 72% showed high correlation to kidney damage, to fibrosis in kidney. And this was highly statistically significant, p-value 10 to the minus of 300. Okay, so we term these tissue unique, kidney unique sites because they have one pattern in kidney and a different pattern in every other tissue. And we asked, well, what happens to these sites when the kidney is damaged, when we have kidney disease? And without a single exception, all of the sites that were low went higher, and all the sites that were high went lower. So they regressed to the mean. They regressed to the mean in the rest of the body, not to some arbitrary line of 0.5. If you take here the extremes, so methylation sites that were at 0.7, so fairly high methylation value, but still lower than the average in the rest of the body, still went up. And vice versa. Let me change this thing to a laser. Sites that were fairly low, so 0.35, if that was still higher than the rest of the body, they went lower. Not only that, the more unique the site was, so the further it was from the pan-tissue average, the stronger this trend was, as you can see here in the correlation. Well, is there any diagnostic value in these things? So if you just take the data from these sites and throw it in a PCA plot, here are your patients. These are the sick people. They automatically pop up. You don't need anything else. Just take this data. You don't know anything about kidney disease, about kidneys. Just throw the data on the plot and you get your sick individuals. We interpret this phenomena as information loss. Why? You have this pattern that is very unique to the kidney, and upon kidney disease,
it becomes more similar to the rest of the body. This unique information gets diluted. Not all the way. It doesn't become like the rest of the tissues. It just takes a few steps in that direction. Okay, so does it have any biological significance? So we looked at DNA areas. If the slide will change, I would look at them. Surrounding these sites, and we saw very strong enrichments in the up and in the down, different transcription factors, and these are transcription factors that are known to be involved in regulating kidney tissue and to be involved in kidney disease, in renal diseases. Not only that, genes associated with these methylation sites also show differential expression between controls and patients with kidney disease. So this looks to be biologically significant. Well, okay, is this just a weird kidney thing, or is this something global that affects other tissues as well? So we took the same huge data set and decided to identify all tissue unique sites. We relaxed the criteria a bit. If you get 100%, you're probably too stringent. And we required that the 15th percentile in the tissue we care about has at least 0.1 difference in beta values from the nearest 5% of all other tissues. And this is a fairly stringent criteria. You can see one example here. And across multiple tissues, some have many such sites, some have none. The one that I'm not showing here and I won't talk about is placenta, that actually has more sites than all other tissues combined. But as you can see, in cartilage, in brain, in colon, in liver, we see thousands of such sites. Are they functionally important? So if we run Homer to detect transcription factors binding these unique sites, we can see that different tissues, they're all enriched for transcription factors, but each one has a unique signature of transcription factors binding these unique sites. Okay, so let's see if this is relevant for other diseases. We took a big data set of liver diseases, and I'm clustering here all diseases. We did not separate them, but these are your usual suspects, non-alcoholic and others. And one of the nice things about this data set that in addition to liver samples from people with liver disease and healthy controls, we also had adipose samples from the same individuals. And now, if we look at the sites, oh, and I forgot to mention the most important thing, the color scheme. So if it's low here, it's blue. If it's higher, it's red, okay? We keep this color scheme throughout this talk. So the blue ones, the ones that are low in the tissue, almost all of them had a positive correlation with liver disease. The unique ones that were high, that's the red ones, almost all of them had a negative correlation with liver disease. And almost none of them had no correlation with liver disease. So without knowing anything about liver or liver diseases, I can pick thousands of sites, and all of them are going to correlate to the disease. And by looking at the methylation pattern, I can tell you which direction the correlation is going. But this is only true in liver. If I take the same sites in the same people and I look in adipose tissue, that's what I get here on the x-axis. I get a smooth distribution across 0, here and here. Nothing special, no two clouds. So information is lost, but it is only lost in the sick organ. Other organs are not affected, or at least adipose in this case. Okay, so we have epigenetic information loss in disease. What about stress? We heard a nice talk about skin earlier, and in the skin you have the upper layer, the epidermis, and the lower layer, the dermis. And upon exposure to the sun, that is a form of stress that is highly related to aging. It's even called photoaging. If we take these unique sites and we do the same PCA, we can see a clear separation between the dermis, here with X's, and the epidermis in circles, and between sun-exposed, here in red, and sun-protected, here in blue, but the separation is nice only in the epidermis, which is the part of the skin that does the photoaging. The effect on the dermis are much more minor. And if we take controls, so some of the controls like random sites, sites with high standard deviation or low standard deviation, some of them can distinguish dermis from epidermis, but they do not distinguish sun exposed from sun protected. So stress here causes the same phenomena, information loss in the skin. So this is just a chart to explain how we should read the next one. So we're keeping the same color scheme, red and blue, and we're looking at the difference between a healthy tissue and a sick tissue. And if the red is on top and the blue is on the bottom, this is the opposite of the correlation one. We get regression to the mean, what we called information loss. And if we get them mixed up, then there's nothing. And if we get the other one, they diverge away from the mean, and we don't know we don't expect it, and we won't know how to explain it. So in the liver, like you've seen previously, almost all red sites are above the line, almost blue sites are below the line. 99.6% of the sites went in the direction we expected, and we saw information loss. In the brain, if we look at autism, our negative control, this is not a health-associated disease. We don't expect to see anything like that. And indeed, the dots are scattered all over the place. We don't see a trend. If we look at type 2 diabetes in adipose tissue, VAT and SAT, the red ones are predominantly above the line. The blue ones are below the line. Information loss. If we go to sun-exposed epidermis, like you've seen previously, most of the red dots are above the line, most of the blue dots are below the line, information loss. But if we go to the dermis and we go to the pancreas, we see the exact opposite. We see divergence away from the mean. I don't know how to explain this. We don't know what it means yet. We haven't been able to figure out why some tissues behave this way and some tissues behave that way. If you have a clue, please let me know. Okay, so what's the functional importance of these sites? So we took all the sites and we did a bunch of analyses, and the one that we like the most is if you take all of these sites and you look at 15 chromatin states, and you look here at about 100 tissue types and cell lines, you can see very strong enrichment only in one place where you have liver enhancers. So these sites, and these are specifically the blue, the low ones, are tissue-specific enhancers. These are enhancers, but they're only marked as enhancers in the liver. They're not marked as enhancers anywhere else. This is the functional importance of these sites, at least the ones that are marked as low. Well, what about aging? So we see the same phenomena in aging brains. We see strong information loss. In colon, we see a very, very strong trend. You can see here the unique sites. They alone enough to organize them according to the age of the samples. Here it's actually divergence. It's not information loss. It's 2 lines because it's actually sampled from 2 different sites in the colon, and that's actually has enough methylation information to separate them. And again, none of the controls are able to arrange them nicely by age, only this thing. And now we ask, okay, so when we age, some tissues may change, or in disease, change the cell composition. Is this reflecting changes in cell composition, as some of the epigenetic clocks show, or do the cells inside of themselves change? Is it within or of cell populations, or both? So in the brain, if we look at tissues, at the tissue in bulk, we see information loss. If we look at FACS-sorted samples, we see in neurons information loss, but in glia, we see divergence. So it's not only changes of the tissue, it's also changes of cells within that tissue. And we also see this in CD4 cells with age, where we see very, very strong regression to the mean with age. And in cells grown in culture, I'm not showing it here, but we also see the same phenomena. So this happens not only by changes of cell population, but within the cells. So Nir Barzilai and others have asked, is aging a disease? And I think if we look at this data from the information theory of aging standpoint, the theory that posits that information loss and specifically epigenetic information loss is a big driver of aging, the resounding answer is yes. But I think here we need to invert the question. This is the wrong question. Multiple diseases are actually organ-specific manifestations of aging. So kidney disease is an old kidney. Liver disease is an old liver. This isn't universal. Autism is not an old brain. But for some diseases, especially those that we see go up exponentially with age, this is the way we want to look at it. And to summarize, we show strong support for the epigenetic information theory of aging. Epigenetic information loss happens in an organ-specific manner for multiple but not all diseases. From the information loss standpoint, aging is a disease. And we can diagnose multiple diseases from methylation patterns alone without any other knowledge of the disease. We also have an hypothesis. And we haven't said here anything about causality, and causality is very challenging with methylation, but we do hypothesize, and we'll need to prove it, that closure of tissue-specific enhancers drives the smoothing of the epigenome and drives aging and disease. And with that, I'd like to thank the people who did the work, predominantly my talented postdoc Naor, and all other members of the lab and the funding agencies, and you for listening, and I'll be happy to take questions.
מהו האיבר שמזדקן הכי מהר?
That's a difficult question. It's really individual. And the studies show that different people have organs at different ages. For many of us, the first thing to fail is the cardiovascular system. So that could be one answer, but I would be hard-pressed to find an organ and say this is the one that ages first. Some people, their mind is gone by 60, others are sharp at 100, as some people here study them.
אז אם אנחנו רוצים לעשות בדיקת גיל, אנחנו נצטרך לעשות את זה איבר-איבר?
So this approach right now, it's not applicable to the clinic as is. No one's going to take brain samples to say what the age of the brain is. We have some preliminary data that some of it is reflected in specific cells of the blood, and maybe from blood samples we'll be able to learn which organs age at which individual. There's also works doing this with proteomics, where you look at specific proteins in the blood and say something about the age of the organ. But it's still at its infancy. Yes, please.
So there was a huge experiment done with brain that checked the specific epigenetic markers for each cell type in an organ, with the concept to say that they could then identify from cell-free DNA in the blood from which organ it comes. But what you say is that these specific sites change with age. So the atlas that they have done should be corrected with the age of the patient.
So
can you repeat the question only?
Yeah, I'll repeat the question. The question was that there were atlases generated, correctly if I'm wrong, about epigenetic marks of different tissue and cell types, and then trying to analyze from cell-free DNA in the blood and working back. And yes, we know the epigenome changes with age, and this is a delicate point because these methods, a lot of times there isn't that much methylation data per organ or per cell type, and people try to do sort of deduction studies, and all the reviewers, they always ask, well, why don't you do the convolution? And you do the convolution on samples, forget my samples, you just take public data sets and you do the convolution analysis, and sometimes it works great, and sometimes it tells you about cells you know are not present in those samples, or cells that are present and shouldn't be there. You do analysis of brain samples and it tells you, well, these samples don't have neurons.
I don't know of brains without neurons. Maybe they sampled politicians, I don't know. So yeah, you always need to be very careful in the way you analyze these things. Yes, please, at the top.
Different patterns of different diseases, different methylation loss patterns in different diseases within the liver?
So within the liver, I don't think we had enough samples to distinguish. For us, it looked like it was all trending in the same direction, but I don't think we had the statistical power to say if the difference were subtle, we probably wouldn't have seen it.
I'm asking specifically the difference between a disease and like an aging. So there's no clear definition.
So in these experiments, they were all well constructed, so there wasn't that much of an age gap, so the controls were clustered tightly enough and the disease. So I know that we see very similar patterns in mouse aging liver, for example, I can tell you that. But going and comparing in the same organ aging and disease, we don't have that yet. That would be a good experiment to have.
Was there any thought given to looking at the mutational signatures involved in these specific tissues, right? There's been work that's been done that's kind of shown that, you know, each tissue is under some unique stresses and exposed to unique things, and how that might also affect the patterns that you're looking at.
We haven't looked at that specifically, but if I had to guess, I would say there would be an overlap because a lot of the mutational burden is on open chromatin, and at least the low ones, they're open and they're only open in that tissue many times. So there's probably just because of that going to be a statistical overlap. Can you use the mic so they'll hear you at home?
Can specific proteins like tau or maybe atherosclerotic plaque drive specific epigenetic changes that you found correlations for?
Probably yes. So it won't be a single loci, it will be a few. But yes, if you deregulate transcription factors or the machinery around it, because the relation between methylation and expression sometimes is bidirectional. So I would expect yes. Thank you very much, but you're not done with me yet. I would like to introduce our next speaker, Noam Maoz from Centaurix, to talk about measuring aging in the community.
Okay, so thank you very much, Elia, for organizing this convention and inviting us. So I spent the first decade of my career in Bar Ilan at the great lab of Professor Chaim Cohen. And I would like to tell you a little bit about what we are doing at Centaurix today. After my postdoc, I was fortunate to join Shimon Meshi and Yitzhak Dershowitz, who are here as the co-founder of the company and CTO. I really hoped after my postdoc that there will be a place, to work in this field, outside of the academy, to push what we are doing in the academy, also the industry, and there was some luck here. Okay, so I will explain a little bit about the company. So we are a privately held company. We were founded in 2018. We develop innovative technologies in longevity. We operate from a state-of-the-art facility in Jerusalem, and we employ over 70 scientists, diverse, from scientific and health disciplines. Now, our main projects are, the first one is about cellular rejuvenation. What we are doing here is taking the epigenetic rejuvenation approach. For the last 6 years, we have been trying to isolate a cocktail of epigenetic modifiers that can rejuvenate the cell, mainly by the epigenetic clock, but also transcriptomics. And I'm happy to say that we have a few patents about these cocktails that can reverse the age of the cells epigenetically by a few decades. At least I think officially I don't know of any better methods, and also the approach that we are taking is safer than what was published until now. Now, in order to have some validation of our success in the cellular rejuvenation project, meaning we're taking cells from old people and trying to rejuvenate them, we need to be able to measure the molecular hallmarks of aging. So we did work on that also, and develop wet processes, technologies, improve current technologies in order to be able to measure those molecular biomarkers at a larger scale, at a lower cost. And so this is kind of relates to the other department of our company, which is aging diagnostics. So it started as a department that supports the cellular rejuvenation team, but then, like we thought about it, there is a much greater need in aging diagnostics than only to support our other department. And what I'm talking about is the situation today at 2024 that we have. This is just a small example of anti-aging treatments that work in rodents. All of them were shown in peer-reviewed papers to extend lifespan, delay aging diseases. But the thing is, okay, we have many candidates, and every year we have more candidates. The question is, when will this work in humans? Like my grandfather asked me 10 years ago, Noam, when is what you are doing will help me in my lifetime? And I told him, yeah, Grandpa, I hope so. But he died, I mean, now, a few years ago. And to tell the truth, even if he was living today, with the current approach, there was nothing to give him. And it's a decade of practically zero progress in what we can give people. And the big problem here is not about, in my opinion, it's not about like methodologies to rejuvenate, about interventions. We have interventions. Of course, we can form better interventions, but we already have many candidates. The question is, how do we validate in human? So the road to practical validation. So the gold standard of validating what we are doing in rodents, because they live short, we are checking whether the intervention increased lifespan or whether we see a delayed onset of aging diseases. Now, the good thing here is a reliable endpoint. But the bad thing when we are discussing human, of course, is that this is very slow. We are talking about decades of waiting. It's very expensive because it's so long. Maybe we can reduce it, to a decade if we are following onset of aging diseases in a very large cohort, but even a decade. And moreover, it's not personalized. So let's say that we did such an experiment for a decade or two, and we found an intervention in humans that can reduce the amount of cancer and dementia by 5%, 10%, something along those lines. And now I, Noam, want to take this treatment. And the thing is, I will take it, and I need to pray every day that a few decades from now, it will help me. How can I know if it helped me? I mean, the fact that there is a cohort that got some, average, statistical significant advantage of this intervention doesn't say that it will work for me. For any drug, it works for 50%, 60%, 70%, and the rest it doesn't work, and there are people that it will even make them worse. So I need to pray that it will work. I need to wait now a decade or two and get maybe a treatment that will maybe extend my life 10%, and I need to pray every day that it works. So we think this is a problem. This is a huge bottleneck. Now if we go to the other extreme, how do we validate those interventions? I think there is a lot of debate here. The other way around is going into a single biomarker of aging, like the other edge. And here we might talk about the epigenetic clock, which is, very popular recently, and for good reasons. There are many papers, connecting it to biological age. So, and here we are talking about a fast and cheap modality. It correlates with aging, this biomarker, and it's a personal result, meaning I can know with my body relatively early if the treatment is changing the epigenetic clock in a favorable manner or not. The problem is it's very limited reliability. How can you be sure you're optimizing the right stuff? Maybe you are making the epigenetic clock better, but the rest of the body is becoming worse. We don't know. I think there is some paper that claim that alcohol consumption to some level rejuvenate the epigenetic clock or correlates with lower epigenetic clock. So there are a lot of confusion about how much can you trust these biomarkers. And the last approach that I want to say, this is the approach that we took, Centarix, is we can call it a multimodal aging biomarker panel. What we are doing here, we are working with biomarkers, but in a very robust way. So we are taking biomarkers, molecular biomarkers, such as the epigenetic clock, but not only. We are using serum, clinical serum analytes markers for a very robust panel to see the health of the organs clinically. We are taking a functional biomarker panel, meaning all of the functions that deteriorate with age, whether it's aerobics, muscle power, flexibility, balance, etc. And we are also doing imaging to understand the body composition, how the organs look. So we are taking all of this big panel from micro to macro, and we are using it to assess health and biological age. And the advantages here, it's still practical. Yes, not as practical as the epigenetic clock alone, but it's still practical. It's very personalized, and it's reliable for judging health. Now, you might ask, okay, let's say that I'm taking an intervention, and I'm using this multimodal approach, and I'm seeing a favorable profile. Yeah, a lot of stuff are improving, and I don't see toxicity or side effects. Does it mean for sure that it will also mean that I will get more lifespan and there will be like a delayed onset of aging diseases? I don't know. We don't know. I hope so. It seems logical. Yes, that if I'm getting healthier, then it will extend my lifespan. We can be sure, but I will say something which might be blasphemy here. Maybe we don't care. Maybe we shouldn't care, because the question is, what are we after? Are we after improved health, more healthy years, or are we after living longer, just living longer? So if I have a panel, a robust panel, a reliable panel for health, and I see that an intervention that worked in mice to extend lifespan, I see that for me, it brings a lot of health benefits with minimal or no side effects. For me, it's good. I want to be healthier. I want to be healthier now. I want to be healthier in a decade. And if an intervention gives me that, according to this reliable multimodal approach, I'm good with that. Now, maybe I will not live longer. Maybe I will die very healthy, but not very, at very old age. I'm okay with that. Okay, now the problem here, let's say the difficulties here are standardizing such a panel. Like, okay, which test are you going to select? You need to be very smart here. And which machines, which protocols. You need to be very careful how you are going to measure those biomarkers, and also calibrate. You need to calibrate it in the population. You need to understand what happens to those biomarkers in the open. When people living their lives without treatment, what is the noise, what is the signal? So with a lot of work that needs to be done here. And we have started doing this work a few years ago. We have started a clinical trial at 2021 under the Helsinki of Hadassah Mount Scopus. And in this clinical trial, so it's a longitudinal multi-year study with hundreds of participants, aged 20 to 80, only healthy people. It was very important for us to make this, to check those biomarkers on people when their health is mainly affected by chronological age, and not because they are obese, and not because they are now with a great sickness and taking medicine, because again, the cohort is not big, so we don't want this noise. And here, regarding the endpoints, we carefully selected a large, diverse panel of multimodal aging biomarkers. We did a measurement at short intervals, so half a year intervals. And all the biomarkers are measured in-house, so they came to us. Some of the biomarkers, the molecular, again, are development that we did. And we also generated a very large biobank of multiple cell types from each time point, and this allows us now, if we want to check a wet biomarker, we can simply unfreeze the samples and see if we see a signal with age. And now, it doesn't move. What happened? And now the big study goal here is to building a biological age ruler. And the important questions in order to do that are, first of all, how detectable are the aging trends that we are going to see for each biomarkers? Understanding the biological noise. So again, we might see that, in the wild, without treatments, we see that telomeres are extended after 6 months or a year. And we need to understand by how much. It's a natural phenomenon of the human body. And the same question for each biomarker. How does it behave? How can we differentiate between noise and signal? Behavior of wet biomarkers, those that you are checking on cells, on different cell types. So people usually use whole blood for such experiments, but we used subpopulation also because we don't know what happens in T cells, in monocytes, etc., neutrophils, what happens in buccal epithelial, all of the cells that we can easily collect from the body. We want to understand specifically what happens there. Maybe it's different than looking at whole blood. We want to understand correlation between different biomarkers and also develop algorithms from composite biomarkers. And here I'm showing you some results, some portion of the results that we got from the test. So what we are seeing here, you are seeing the population of the study, even not all of them. You see the population, the x-axis is the age from 20 to 80, and the y-axis is the biomarker. And what you can see here is a very clear trend, whether it's visceral fat, balance, working memory, visual memory, a lot of cognitive skills, systolic pressure, CPET. And the interesting thing here, it's a relatively small cohort, very diverse ethnicity, different lifestyle, and we see the aging phenotype so clearly. And we have dozens and dozens and dozens of such graphs of biomarkers. And what we are seeing here, so again, we are measuring from the molecular to the serum to the functional to the imaging. And what we are seeing actually is the phenotypes of a disease called aging. These are phenotypes of a disease. And now the question is, can we take the treatment, can we give somebody a treatment and then follow those biomarkers as endpoints? I mean, if we know that there is a decline, and if we see that the intervention is improving the decline, okay, yes, is reversing the decline on many criteria in this panel, maybe we should use that, because this is practical, this is now, this will yield results relatively fast. Yeah, it's very important, to have this ability to measure quickly. Okay, so based on this clinical trial, we have recently opened a clinic that does this commercially at Jerusalem. And in this clinic, we are doing this multimodal approach. And now I will shamelessly advertise us a little bit. So basically, we are offering biological age measurements at scale. In terms of businesses, we are offering also remote longevity testing because, as I said, we developed biomarkers of aging that you can check on all sorts of cells. And also, we want our clinic that does the multimodal approach to be a site of clinical trials. Like if somebody has some kind of a treatments, supplements or whatever, things that might work in human to delay aging, we want to be a site for this and check it in our physical site. And regarding customers, we have started servicing customers as an advanced longevity clinic. I think the most advanced in the world, where you are taking the multimodal approach measurement, and then we are working with you, with a team of professionals, to rejuvenate those biomarkers, because those biomarkers are very robust from the molecular to the imaging. And thank you very much.
And can you go to accelerate aging? Can you know also how to accelerate aging?
Yeah, I think it's very easy. We can accelerate your aging very fast in 6 months. Just let me give you a recipe and follow it closely. It will involve drinking a lot of alcohol, not sleeping well, I don't know, a lot of stress. Smoking, maybe a war, maybe a small war with surrounding countries. Yeah, it's very...
No, because it can help in your experiments. I mean, it's not so ethical, but it can help in the experiments.
The other side, you mean?
To move,
Can we
move people on both directions.
Hi, Noam. As part of the cohort, I was surprised that you don't take the microbiome, which is generally blamed for everything, including aging.
You're talking about the?
Microbiome.
Microbiome. Yeah, so the question is why aren't we, because she's a participant, we have a few participants of our clinical trial here, why aren't we taking the microbiome? And it's a question about, you can think about it, you know, when we use imaging, why are we using this and not that? And in the molecular, why are we checking this and not that? The question, the thing is that there are a few modalities. There are the molecular, the serum, the functional, and the imaging. And we want a good representation in each modality. Now, we can't take everything. You cannot take everything. But what we know, all of those biomarkers are very interconnected. There is a lot of crosstalk between all of the molecular. So, for example, if you are, you know, remember the paper of Lopez-Outin, the last one of the 12 hallmarks. So you have like the primary, and then the integrative. Okay, so, and the microbiome is at the top. It's affected, but all of the molecular biomarkers below it. And we are measuring some of it below, some in the middle, inflammation. We think we are covering it enough. We are covering the molecular enough, the blood serum enough, the functional enough, very thoroughly, and also the imaging. So you are getting a very robust image of your health. And if there is an intervention that improves biological age, and we are not seeing it in such a, you know, a robust multimodal panel, I'm sorry, it's not there. It's not there. You don't have, you haven't improved health in any meaningful way. Even if you improved it in a significant way in a biomarker that we don't measure, we should have seen the side effects in all of the rest.
Yeah, I understand. So you have selected enough biomarkers to have a complete image.
Exactly.
Another question maybe, will it be for rich people only, this kind of clinics?
So sadly, it's not cheap. It's not cheap, but at the setup of a clinical trial, it will be cheaper. But again, what we are doing here, again, everything is not cheap initially. If you talk about, you know, the cell phones of 30 years ago, it was huge boxes. It didn't work well. It cost a lot. So yes, this is an early thing. We think there is a lot of potential here. We think like this is the way to go. If we want results, like we don't want to meet here 10 years ago. I really hope that you will make a convention 10 years ago. All of us will be here, and we will see not only 25 interventions in mice, like I showed you, but 2,000. And in humans, nothing. We don't want this. Yeah.
All the industry of well-being is subjective. You know probably the studies about happiness, about emotional well-being, and probably it's like a killer app. Let's say a person drinking, smoking, but he's happy.
Mm-hmm.
But he's living well. He's doing whatever he likes, and he feels okay with this. And he lives longer. This is something which is not going into all this data information, and nobody takes it in consideration, because the influence of a person on himself is the highest placebo. So this is something which I think you should intervene in your
Okay,
study.
so first of all, we are taking this into account. What I haven't showed, like in this multimodal, it's also many questionnaires that are, you know, we have a psychologist working with us, so we are probing the human, you know, psyche. The thing is that even if you are stressed, even if you are, you know, there is the duality theory that we have a soul and we have a body and they are connected through some Bluetooth or whatever. So, you know, the mind affects...
It's even more complicated.
Yeah. The minds affect the body. Like if you are stressed, we want to see cortisol level up. We want to see inflammation. We want to see visceral fat. So if you are stressed and we are checking your entire body with this multimodal approach, everything is perfect. I'm sorry, you are fine.
Yeah, but you know, I go to my father, he's 86, and he's going to the doctors and he said, listen, I don't feel well. And they check his data in 86, and they said, listen, you are compared to the average people in your age, you are perfectly, I wish I would be in your position. And this is, what kind of doctor is this? What kind of medicine is this that is not able to analyze my situation based on my specific data, but on an average? You know, you compare to the average. On the average, he's healthy, but from his perspective and the way he lives today, he feels sick. He feels that he's tired, doesn't have enough energy. And there is a solution for him, I'm sure, but not in the conventional medicine.
I'm not sure if you are saying he's 80-something and he's healthy. According to his age cohort, 80-something is not healthy. He's very, very sick. Even if you have no disease, like no conventional disease, at 80-something, you are very, very sick. If a person aged 20 will wake up someday, once he's 20, and he's waking up as an 80-year-old guy, he will be hospitalized with a deadly something that he caught. It's not, and I will tell you something that, I'm not talking about 80-something, like we got like hundreds of people with this multimodal approach. Almost everybody has many problems. They don't have a clue. They don't have a clue. Once you are taking like the biological age and looking at it as a very large puzzle, and you are showing the person everything, people are shocked what they find, like how much they are deteriorated in stuff they never even thought to measure. So the first step is to measure, and then you see how much, even at age 40 and 50, you are deteriorated in some stuff.
Yeah, but the blood test they do, conventional medicine, is not even close to understand.
It's no, yeah, what we are doing now is much more than conventional medicine, and I hope one day conventional medicine will meet us. Until then, it will be private.
Yes, okay, thank you.
Hi. Did you check the nutrition of your participant and how it's affect on the results?
So we check nutrition in questionnaires. We check nutrition with questionnaires, and we have like a lot of blood tests that are, related to nutrition. Also, we have we are doing DEXA scans for body composition. So we have some knowledge about what you eat and how you look and how you function. But this is something that is improved. So, for example, now at the most updated version of of what we are doing, we are using, for example, CGMs, which give us more understanding of, for example, of the glucose, what you eat, how it affects your glucose level. So, again, we are incorporating new stuff along the lines, and with nutrition, we are taking this into account. Yeah. Okay, thank you very much. Okay, so our next speaker is Gil Atzmon from Longevity AI-powered longitudinal longevity studies, and this will be online, according to what I read here.
Thank you for the invitation to this convention, Ilya. Great honor to be here. Great to follow a great company like Centerix. Today, I want to share with you a little bit of what we're doing here at Longevity AI. I'll start with something classic of how we view the world of longevity. I'll do this quick because I guess this is the audience. I'll share then some about the algorithm, but what I'm most excited to share about, which will be the second part, is how we're scaling longevity today with private and very soon public clinics in North America. Okay. I'll start by sharing the screen. And if anybody has a question, even in the middle, feel free. The best thing is to make it a conversation, in my opinion. So a little quick recap of how we view the world of longevity. So we all know the world is getting older, and it's happening at an accelerated rate. We know that our unhealthy years are growing, and they don't match how much medicine has advanced our lifespan. When we look at the data from the CDC in the United States, we see that over 80% of unhealthy years are the chronic aging conditions, which are all of us are familiar with. But what we also saw is that we're actually looking at longitudinal data, and this we're doing with Maccabi and Clalit, and also with the North American partners, we're seeing that the biomarkers are detectably deteriorating even 20 years before we can see the condition. And this should not surprise anybody here. Our take on it is always starting with conventional medicine and then improve from there. So when we asked our data partners, how would they suggest looking at the biggest killer of the heart? They said the Framingham score. But with the same breath, they said, yeah, well, it's the golden standard learned in medicine school. We don't trust it. So I asked them why. They said, well, done 50 years ago on one village population in the States, half smokers, not ethnically or gender diverse. And this is still the way to assess the heart, according to modern medicine, for everybody in this convention. So we said, okay, how about instead of diabetes, yay or nay, like Dr. Maoz before me shared about continuous glucose monitoring, that's indeed the way. The approach is to build models with more information and therefore reduce the error range. So from the Framingham score, which was more wide, now we have more predictability and accuracy to understand what delta in which biomarker can provide extra health years. And this we did for all the bodily systems. So we have for cardiovascular disease, we calibrated for age. So it's the heart age and then the metabolic age for diabetes and obesity, immune age, cognition age. And this is for us just the first half of the puzzle, right? The second half is how can we actually get people to change their minds? Because until we're becoming a mouse, at this particular moment in time, this is the best that we have. We have, of course, GLP-1 drugs and others, but there's no doubt that healthy lifestyle and changing how we behave and even taking drugs regularly is a behavioral change. So we do the best fit between biomarkers that we want to influence and the healthy habit that we think there's a good chance that person would like to do. So we're building it on top of EMRs, genetic labs, biomarkers, and on the other hand, information from the wearable, from Apple Health, Google Health, that connects in turn to Nike, Headspace, etc. And we see previous behavior and trends and what could be a great fit. And of course, the impact to us in the convention is obvious. If you intervene early, you can detect and prevent. We have 3 products, and this is how we deliver longevity at scale. The first one is the algorithm. The second one is a dashboard for medical teams, now live in Dubai, Canada, and the United States, and the app for the members of those clinics. Okay, so I want to show you just a little bit about the algorithm, and then the title of the talk is how do we deliver it at scale. So I'd like to show you the dashboard for medical teams. Okay, so we talked to experts in their field and in the aging conditions in behavioral science, and we asked them, how can we have one conversation? You all study different methodologies, but we want to have one conversation regarding healthy years and health span. So, you know, they said, let's look at all the incoming data. These are all the blood biomarkers that were ever connected by evidence-based medicine to the aging conditions. These are all the genes, starting from the 75 ACMG genes, American College of Genetics and Genomics, clinically valid across the world, but also whole genomic sequencing for clinics who charge, you know, $20,000, $25,000 a year, not in the public system. Wearables, we work with Apple and Google and Samsung to get raw data from the devices. We understand the error range per metric, so we can draw trend graphs which are statistically significant on evidence-based medicine biomarkers, bringing them to the physicians. Usually, it's a big differential for them in the system. Social determinants of health, everything which is not in my control, my physician's control, but I should really do the 150 minutes suggested by the CDC to prevent my diabetes because I'm pre-diabetic, let's say, for example. But if I don't have a gym next to me, or if I never went to the gym from what I can see from my phone, from my watch, this has very little value to give me this advice. And most importantly, behaviorally, if I say no to the first advice I get from my physician, the chances that I will listen to the second one drop significantly. So the behavioral element of what is a great suggestion for me right from the top is super important. So maybe I did walk 8,000 steps a day, and now I walk 4,000. And according to the Framingham or the Framingham Pro that we produce, this can be half the risk. Let's go back to a habit I just used to have. So let's take this walk, but not today because it's raining. Epigenetics, clinical questionnaire, physical examination, microbiome, medical procedures, and my personal favorite engagement. I'm a geek of behavioral science and data science. My previous position, I led the AI department in a behavioral tech, advertising tech company called IronSource. So my job was to look at billions of people, how they use their devices on a daily basis, and just predict what we can convince them to do next. So sorry for other advertisement I have tailored to you, but now I want to tailor you something which is for the best of your health span. On the right-hand side, we talk to experts on lifestyle. How can we change them in reality? So everything regarding activity, nutrition, sleep, mind, rhythm, even down to pharmaceuticals. So in the center is what I'm most excited about a data science geek is how can we predict and prevent the conditions of aging. And just like the Framingham score, also the American Diabetes Association is a global standard. But, you know, it's not perfect. Also, here's diabetes, it's pre-diabetes, yay or nay, for example. But what we can do is we can take all the evidence-based medicine ever written from PubMed, filter everything which is not AAA, like not published on journals like Nature, not recreated in the last 10 years, or not written by authors of clinical procedures known to that country, like North America is our focus. So instead of one paper, like the Framingham score, we can do something which is more tailored to the person in front of us, if we're physicians, to the age, gender, and ethnicity of people in front of us. And chief medical officers usually say, okay, I like that because all of the clinical procedures that I use in my clinic and study in medicine school are available in this knowledge graph. And when we're doing our AI, which is predicting the risk and which is giving the result of what I can do with it, usually what happens is that it takes from things that they already know, and if not, it's from a box of evidence-based medicine that you saw. Every edge in the graph is a paper there. So they know that all the results always have reference to evidence-based medicine, and this is an example of something I click in the graph. Okay, I'm putting this aside. Some of you who we met already probably saw this. This is something a bit new. This is from recent months and from the clinics that we've launched. So this is how we welcome people into the clinic. I'll give you an example from our design, and then I'll give you an actual demo user from one of our clients. So let's welcome John in the clinic. We can tell him, hey, John, we're happy with your sleep. We're a bit concerned with your activity levels. From all the body systems, you know, the heart is aging the fastest comparing to people of age, gender, ethnicity. Here are your needle movers. They're here because they make the most impact, but they're modifiable. And here are things that you already love doing. Let's just choose one of them. And it can be one of the most significant things that you can do for your health today. Now, let's go to the live system. And I'll show you a demo user that we prepared with one of the clients. By the way, if anybody has a question in the meantime, please feel free. Okay, so John here. So we see how the classic risks are calibrated for age. We can see the aging conditions and their leading biomarkers. If the physician doesn't have time, if they just have, you know, that 1 minute, here's the combination of things that are very good for that person's health, evidence-based medicine, and also something that matches what they love doing. Cool feature, and now going to be released out of beta, is that we can have all of these biomarkers in very little time, because that's the challenge, right? In longevity clinics, when we meet physicians, they tell us, well, I love my profession, but I wish I didn't have to work 2 hours of paperwork before every patient. And I wish I could accept not 2 patients a day, I could accept 8 patients a day. So we take automatic information from electronic medical records, or you can just drag and drop a PDF of a vendor we're working with. Well Health is one of our clients. They serve every third doctor in Canada, have a few hundred clinics in the US. And you can see that Florence, our generative AI, can take every piece of information from an approved vendor. And if we have vendors in the audience, which is basically a lot of longevity companies who take a specific piece of information, have a specific set of results, and want to offer them to clinics. So our generative AI can look at the results and analyze what is very important for that specific individual to eventually contribute to that biological age and to that longevity plan. And here you can see the results from this one. And this is a genetic example. And what physicians are usually most excited about is they can get that book about you. So each of us has lots of information on a setting of longevity clinic. We also do public health systems now, but it's hundreds of pages of PDFs. And in under 10 seconds, we produce a book about that person in front of us. And we can see a letter from the physician. We can see a bit about the hallmarks of aging. Doctors usually take around 10 minutes to review it instead of these 2 hours. And now with a clinic that we deployed in North America, they just scaled from 2 patients a day to 8 patients a day. The chief medicals are happy, the chief financials are happy. This is the longevity plan. Again, this is to fine tune together with the patient, nothing like doing joint work. This is all the medical history summarized, body composition, visual, where to focus the health efforts, which mainly is the risks, starting from the medical standards, but also building up everything evidence-based medicine has to offer today. And unsurprisingly, there has been some advancement in recent decades, but they've not made their official indexes, but we include them here. And we have, of course, the biological ages, which is calibration for age, the lab results. Okay, there's more to show. We do a little bit more. We also have an app, but in the sake of time, I'll pause here, and if anybody has any questions. And if not, please, as a longevity company, feel free to get in touch. Maybe one of our clinics would like to work with you. If you want to be a patient and you're in North America, please ping us. We'll direct you to great professionals. Okay, if anybody has any questions.
You started your work saying that you can do more accurate prediction than Framingham experiment. So could you say something about the accuracy of your prediction compared to the previous predictions? Like you started with the heart, so could you say something? Because I know somehow I remember the number of 8 years standard deviation for the current genetic clocks. So I want to get some numbers.
Of course. So I'm a data guy as well. But just to frame what we're doing, and your question is super important, it's the heart of our studies. But I want us to frame what clinics want today in the public system in North America and the opportunity to improve that indices. What clinics seems to want today, and this is where we've taken our product, is the clinical golden standards, the Framingham score. Like it a little bit less. This is what the boards, the medical boards in those countries want to see. And what we're doing, we're curating, or in Hebrew, we're otzrim avuram, like a museum, the biomarkers that have evidence-based medicine next to them. And they consider it a lot of value, and they also consider it evidence-based medicine. In parallel, we're working with our data partners to develop these indices, the index of the heart, of the risk. Once we publish that, I'll be happy to ping you. If you leave me your name, I'll be happy to let you know. If you want to talk offline what we're doing so far, I'll be happy to share on that as well. But we currently have not published or using any of the new indices.
You said that the business model is B2B. You go with clinics, but what about individual or business or let's say I have let's say a business center, 100 people work there, like we work, and I wanted to have tools to help people be healthy while making business. So I have different needs. When you go to clinical places, you take in advance that people are sick. We want to take care of healthy people that want to improve their performance, okay? It's a little bit different than the target you took in your business. So what do you have any product, you have any ideas about this market of working with healthy people that want to have better performance, not just people that want to live longer or be more healthy?
Okay, great question. Yeah, sorry.
No, it's a difference between being healthy and live longer and being perform good and make good business, for instance. Okay?
Yeah.
Where do you take it from? This is what I'm trying to understand.
Okay, so great question. I completely agree with you, and we seem to share the same vision of improving performance and health, also in healthy people. And by the way, shout out to Arya next to you. Hi. So, you know, what we see from our clinics that they're serving mostly generally healthy people. Even if they have one of the chronic conditions of aging, the clinic and the individuals, we call them members, not patients, just for the same reason that you mentioned. They don't consider themselves sick. And we want to predict and prevent other chronic conditions that had not yet manifested. An interesting way that is not only B2B2C what we're doing, we're B2B2C because we're also developing the mobile app and help navigate that end user experience. But there's more models. Our clinic in Dubai and our partner in Canada also plan to use it for employers because you can automate that report, and some of it does not need a doctor, so there's a version of it that can be automatically produced. We don't deliver it directly to consumers, but employers can just print out thousands of those in under a minute for all their employees. If you have any other suggested business model, I'll be happy to discuss with you. But generally speaking, I think I really agree with your approach. There's so much potential with healthy people. One more note on that. Our partners in North America really care about delivering it to that annual checkup with the physician, with my primary care doctor. It's the parallel in Israel of going to your doctor and say, hey, take a general checkup on me, please, which I assume many in this conference like myself are doing. But there it's like a setting that's covered by the insurance. And in there, you're treating generally healthy people without a specific condition once a year, giving them an overview about yourself. And this is also one of the users of the system. I hope that answers your questions. If not, I'm happy to continue offline.
Okay, thank you very much.
Thank you, everybody. Great conference.
We're starting our last session for today. Into the clinic, what can be offered now and in the near future. Those online, okay. We have quite many talks for today. So I ask all speakers, including online speakers, to keep the time and to start and to end in time. So our first speaker is Dr. Marius Kyriazis from National Gerontology Center, Cyprus, intermittent fasting, hormesis, and adaptive responses. Please.
Yes, I'm here. Hello, everybody.
Oh, nice.
You can see, okay, I think. The slot just after lunch is the worst slot that any speaker can have, and I seem to get every time this slot, so I don't know why. Hopefully I'll interest you and stay awake. I'm going to talk about intermittent fasting, hormesis, and adaptive responses. when we talk about dietary restriction, what it is basically is a kind of stress for the organism, mild stress, with the aim to upregulate any damage and repair damage that seems to be present due to aging or due to other causes. So the general idea is to cause mild stress to the body, and so increase resilience and improve health and function. So one of these kind of stresses, positive stress, is intermittent fasting. This is a positive nutritional test, and in general terms, is a good thing to have. But this stress has to be within certain limits. We cannot have too much of it or too little of it. And here is when the concept of hormesis comes in. Hormesis is found in many biological areas. Basically what it is exposure to mild stress or a toxin or a damaging event, but it has to be mild, stimulates the adaptive beneficial effects for the body, the cellular stress response, and so the body becomes stronger. The biological mechanisms involved in this hormetic phenomenon include improvement of antioxidant activity, modulation of autophagy and apoptosis. And I talk about modulation because it is not increasing it or decreasing it. If it's too high, it increases this. If it's too low, it increases it. So that's the modulation aspect of autophagy and apoptosis. And other beneficial effects like cell signaling and improved function of the gut microbiota. So as a result of this mild exposure to damage, we have improvement of different biological areas, and the organism becomes better able to cope with a more stressful event in the future. You can see from the image here, if there is no stress to start off with, and then we are exposed to sudden stress, then there is cellular damage. But if we have the hormetic response before, in other words, if we train our body to respond well to the damaging effect, then there is survival of the cell and better health. Examples of hormesis are physical exercises. When we go to the gym and we do exercises, basically what we do is put our muscles and skeletal system under mild stress. And this makes us stronger. The same with brain exercises, keeping our brain active with mental exercises. We have nutritional stress, nutritional hormesis like calorie restriction, intermittent fasting, and exposure to physical agents, mild heat like sauna or cold, or even mild poisons like arsenic. All of these have been shown to have beneficial effects for the organism if they are within the hormetic window. And we even have social hormesis when we take part in the social environment around us, or sexual hormesis, which may upregulate several sexual hormones and other beneficial effects of the brain and on the body. When we talk about intermittent, this is a general term, not only for intermittent fasting, but it is a term which shows that the stress, the exposure to the stimulus, is not regular or monotonous, it's changing all the time. And this can be applied not only in the nutritional area, but also in other aspects of our life. So in general, it's a good idea to advise people to lead an intermittent exposure to test to any bad stimuli, like intermittent exercise, sometimes too much, sometimes too little, sometimes medium, intermittent fasting, intermittent exposure to cognitive stimulation, and so on. Intermittent is good because it activates hormesis. when we come to intermittent fasting, there are different ways of using this technique. I'm not going to go into big details, but some people fast for 2 days in a week and eat normally the rest of the time. Others fast every other day. Others fast for 16 hours, fast without eating anything, with water only, and 8 hours they can eat as normal. It doesn't mean that they eat for 8 hours, but they eat as normal. And this creates a mild stress to the body. So it stimulates the body to have resources from reproduction to somatic maintenance. In other words, making us stronger and healthier. But apart from the action itself, apart from doing the intermittent fasting, the ingredients of the diet are relevant too. So we have a combination of the practice of intermittent fasting plus choosing to have certain ingredients which act in a hormetic way to stimulate cellular stress response. And examples I mentioned here, for example, dark chocolate, spices. We have the resveratrol, well known from red grapes and berries, and several other ingredients which may be beneficial for somebody who is practicing intermittent fasting. Now, for people who do not want to fast, the same we have with calorie restriction. There are people who do not want to follow a calorie restriction diet. We have the mimetics, which are compounds, chemicals, or nutrients, or molecules, which mimic the biological and biochemical effects of the calorie restriction or intermittent fasting. So during intermittent fasting, we have activation of certain pathways, which same pathways may be activated by nutrients. So people who do not want to do the diet, they can choose, in theory I'm talking, they can choose these mimetics, which have similar effects as the action itself. And the most well-studied are resveratrol, oxaloacetate, deoxyglucose. And you can see here, there are exercise mimetics as well. So people who do not want to exercise, they may choose certain chemicals which mimic the effects of exercise. This is just to show you that biological actions are universal. It can be activated by the action itself, by the lifestyle, or through chemicals or nutrients. And in theory, they have the same effects. They have improvement of lifespan, health span, and prevention of diseases. And I will show you these examples here to show that there are many calorie restriction mimetics. I'm not going to go through everything, but just to show that there is intense research in this area. And maybe people who want to look into this in more detail and see and use combinations of these mimetics like rapamycin and metformin, they could get the same effects as the calorie restriction or the intermittent fasting itself. And before I move on, I'll just tell you that there are not only nutritional mimetics, but also exercise mimetics I mentioned before. You may get some of the effects of some of the benefits of exercise just by taking certain nutrients. And they are also sex mimetics. We did some research showing that during sex, there is production of certain chemicals and certain biochemical situations, which can be mimicked by taking a number of tablets. So you get the same benefit of the organism as the action, in this case, the sexual action itself. But intermittent fasting has a beneficial effect on the gut microbiota. We know that the gut microbiome has many effects on health, and its function diminishes with aging. So it's important to keep it in good health. And it also affects, in my opinion, most importantly, the brain and the function of the brain. So it's important to have it in good state. So intermittent fasting, if done properly, can stimulate the function of microbiota, and that makes the bacteria in our intestines produce certain metabolites which act in a hormetic way. So we have a double effect here. These metabolites stimulate the organism to respond to the challenge and create improved adaptation and robustness. Now we come to something quite important, because it's something that not many people or anti-aging specialists or biogerontologists discuss, is our environment, the digital environment. We all live, and the example is just now, you all sit there in your screens or in the theater, listening to me in a digital way. You don't see me as a person. So all this digital information goes to your brain. The neuron cells, the neurons have to assimilate this information. And I'm talking about information that requires action, not trivial information. So important information has to be assimilated in our brain. And intermittent fasting helps in this respect. Improves, for example, BDNF and other neurotrophic factors. Stabilizes insulin, and this plays a role in as source of energy during demanding cognitive tasks. And intermittent fasting also reduces oxidative stress in the brain. So the combination of intermittent fasting and living in a digital society with information that makes us act is something that we have to study more. And the reason I'm saying this is that if you think about it, when we do intermittent fasting, we are stimulating the organism to respond to the stress and activate certain biochemical pathways which are beneficial. But if at the same time we live in a cognitive, digital, technological society, this by itself stimulates the brain to create the same chemicals or compounds. And in this way, we have double the stimulation. So we have two good stimulations, the intermittent fasting, which is good, and the exposure to digital information, which is seen as a challenging stimulus by the brain, which is still good. But the addition of these two good stresses, maybe it will give us a negative situation, because it will be too much. In other words, we'll have too much stimulation by doing two good things. We have to be careful with that, to not overshoot the therapeutic hormetic window and stay within the beneficial effect. And the general way to think about this is by seeing whether you feel comfortable or uncomfortable with the stimulation. If you feel comfortable with any stimulation, it means that it is good and hormetic and adaptive. If you feel uncomfortable, it means it's too much and you should stop it. So in conclusion, we've seen that intermittent fasting should be within hormetic principles. In other words, not too much and not too little, but within a certain limit, improves adaptation to stress. And there are different biological mechanisms like modulation of autophagy and DNA repair and so on. And we have the mimetics of intermittent fasting, compounds or chemicals which reproduce or mimic some of the physiological effects of the action. And the combination of intermittent fasting with exposure to the technological environment should be done with care in order not to overshoot the beneficial window that is adaptive for ourselves. And this is, in general terms, what I was going to say. And here is me practicing intermittent fasting using a digital device. In other words, reinforcing what I told you, that everything should be in moderation. Even if you do two good things, they don't always amount to one good thing. They may amount to a negative thing. And thank you very much.
Thank you, Mares. Any questions?
I have a question about prolonged fasting. Does prolonged fasting has significant higher effect of what you was talking over the intermittent fasting? And second question, if there are some biofeedback devices and options for biofeedback devices which can track the effect of intermittent and prolonged fasting to give person alert that fasting came to some stage which is dangerous.
We cannot give individual advice for every person. Some people may get a lot of benefit by prolonged fasting. Others may not. For example, myself, I cannot do prolonged fasting. If I do prolonged fasting, I'll get negative effects. It depends on your own personality and on your own organism. And this you can judge from your own feelings. If you do prolonged fasting and you feel okay with it, it means that it's good for you. If you feel very uncomfortable and unwell and vomiting and have headaches, it means that it's not good for you, and you should do a less prolonged period. Biofeedback devices there exist, but I don't trust them too much because there are so many effects that change in our body that cannot be measured by a single device. So basically, it's an individual advice, individual person, and also it could be okay for you for many weeks, and then you'll find suddenly that if you continue doing prolonged fasting, that it will not suit you. We change with time. So I advise personally to see what works for you.
More questions?
Do you have any data or knowledge about exposure to low level of radiation or energy? And if it also creates the positive effect on the body? It can be from sunscreen to sun exposure, ultrasound, even low level of radiation. Is there any data about it?
Yes. In fact, the whole chapter of Hormesis started with the study of radiation, the effects of radiation on the body. And there are many, many studies which show that certain low-level types of radiation are beneficial to the body. There is quite a lot of discussion about X-rays and MRIs and so on, whether they are beneficial or not. But up to certain limits, including electromagnetic radiation, radiation emitted from laptops or mobile phones, if it is within a certain limit, then it's beneficial. It's better to have it than not to have it at all. And if you are interested, there are many studies which set the limits for this radiation. It's a complex area, but basically any type of stimulation within a certain limit is good, including radiation, including poisons.
Thank you, Maria, for your interesting report. Good luck. Now we are moving to the next speaker, Dr. Mario Krause. He will talk about if you are looking for longevity, don't die from cancer. Nice advice.
So, hello to the audience. Hello
Ilya, all my colleagues. Wonderful to join this wonderful conference second time. I'm really be happy that for the invitation. And yes, I will try to give some informations about the same story I mentioned last year, but maybe with some different remarks on it. So let me see if I can share. Well, so far I hope you can see the screen. So the talk is, if you are looking for longevity, don't die from cancer. And as you can see here, cancer is still one of the top killers, for example, here in Europe, where it brings 26% of all deaths in 2018. So there's a German Center for Personalized Prevention and Optimization is using a new approach for early detection of cancer, and this presentation wants to explain the setting up. The earlier a cancer is detected, the more treatment options there are, so more lives can be saved. The European Union has recognized the need of early detection, as you can see in the presented statement from 2022. Early detection of tumors significantly improves the survival rate. The 5-year survival rate increases dramatically if cancer is found in the early stages of tumor development. This can be postulated for all cancers. So cancer screening is highly recommended, and there are various ways to screen for cancer early out there, such as mammography or colonoscopies or various ELISA tests. However, none of these is suitable for universal cancer screening due to high price or limitation on single tumor entities or insufficient sensitivities or specificities. Approximately 300 different types and subtypes of cancer are known worldwide, which vary greatly in occurrence, frequency, and consequences. However, in Germany, there are only 5 types of cancer for which regular screening examinations are currently reimbursed by the health insurance funds. For example, breast cancer, which counts for 14.5% of new cancer cases annually, prostate cancer, 12.8%, and you see colon cancer, skin melanoma, and cervical cancer. These 5 types of cancer account for about 45% of the approximately 500,000 new cases of cancer in Germany each year. For the remaining 55%, there are currently no statutory measures for early detection. If you assume that all people would regularly take advantage of screening offers and that all examinations would work flawlessly, more than 250,000 new cases of cancer would remain undetected in this country every year due to a lack of symptoms and a lack of early detection methods. While the usual approach involves developing a specific screening test for each individual type of cancer, the question arose as to whether there were commonalities that could be used to identify cancer cells. This is where the findings on the metabolism of cancer cells came into play. If it were possible to identify the specific metabolic changes, it would be possible to distinguish between a sick person and a healthy person. This in turn would initiate more detailed diagnostics. Cancer screening is a crucial aspect of modern healthcare. To achieve effective early detection, we need a method that can ideally identify all types of tumors. This universal approach should not only detect various tumors, but also meet several other important criteria. Firstly, the method should demonstrate good sensitivity, meaning it should reliably detect as many tumors as possible. While high sensitivity is important, it's not the sole determining factor. Even a sensitivity of 50% would be a significant improvement over having no screening at all. This logic was applied, for example, when introducing prostate palpation as a standard screening procedure, despite its ability to detect only about 1/3 of tumors. Specificity is perhaps even more critical. This measure indicates how many false positive results are generated. In cancer screening, where the incidence of the disease is relatively low, it's crucial to minimize false positives. Too many false positives can lead to unnecessary stress and further testing for healthy individuals. The ideal screening method should also be able to distinguish between benign and malignant tumors. While detecting precancerous lesions is important, it carries the risk of overdiagnosis and overtreatment. This is less problematic for easily accessible areas like the skin or colon, but becomes more challenging for internal organs. Current imaging techniques such as CT, ultrasound, and MRI have limitations in differentiating between benign and malignant tumors. So biopsies can provide more definite answers, but they are not always possible or practical. And in some cases, monitoring tumor growth over time is necessary, which can be stressful for patients. In conclusion, the ideal universal cancer screening method should be highly sensitive and specific, able to distinguish between benign and malignant tumors, and practical for use across various organ systems. So what about changing the strategy? So this is when a multi-cancer filter test in combination with PET-CT was born. So the idea was a multi-cancer filter to allow the targeted use of imaging technologies. The criteria is to meet universal, sensitive, true early detection, specific, and affordable. If we are talking about cancers as malignant transformation of cells, how can we assess malignancy? We can look for growth rate, growth behavior, cell differentiation, invasive growth, and metastasis. In 1995, the gene encoding transketolase like 1 TKTL1, a unique derivative of the enzyme family of sugar metabolizing transketolases, was discovered by Johannes Koy at the German Cancer Research Center in Heidelberg as part of a genome analysis project initiated by later Nobel laureate Harald Zuhausen. Already in their first description of the gene, its transcripts, Koy and colleagues suggested that the protein may have altered enzymatic activities and may form a heterodimer with the normal transketolase TKT. In 2019, a Chinese study confirmed the presence of TKTL1-TKT heterodimer as well as the altered enzymatic activity caused by TKTL1. The altered enzymatic activity leads to a strong increase of ribose, as you see, and the essential building block for DNA. Thereby, TKTL1 enables and controls the DNA duplication and the cell cycle. The acetyl-CoA production of TKTL1 created a previous unknown metabolic link between pentose phosphate pathway and lipid synthesis. TKTL1 directly triggers stages 1 to 2 of malignant transformation by increased sugar metabolism. Prior to mitosis, the TKTL1-induced sugar metabolism does not only provide ribose as essential building block for DNA synthesis. By providing acetyl-CoA, it even enables the effective conversion of sugars to lipids or fatty acids for the construction of cell and organelle membranes. The exact processes were presented by Li in 2019 in the Journal of Nature Communications. The theory developed by Li shows, however, that TKTL1 is expressed first, followed by the transformation of a heterodimer from TKTL1-TKT, which significantly accumulates ribose 5-phosphate, thereby triggering the cell cycle. This push effect, TKTL1-induced increase of ribose 5-phosphate concentration, controls the synthesis of new DNA. TKTL1 expression directly or indirectly triggers all stages of the development of a tumor from a benign stage to an invasive malignant stage. Stages 1, the accelerated tumor growth, and 2, displacement of the healthy tissue, are triggered directly through TKTL1-induced increased sugar metabolism. Stages 3, 4, and 5 are triggered indirectly through the creation of an acidic pH in the tumor milieu by lactate excretion as a metabolic byproduct of TKTL1 metabolism. The expression of TKTL1 initiates diverse stages of malignancy progression. The entire process, starting from TKTL1 expression in individual areas of the tumor tissue to metastatic disease, is initially slow and hardly noticeable. With the enlargement and multiplication of TKTL1 expressing tumor areas, the process gains momentum, and it is progressive, and there's no turning back. Finding a biomarker given evidence of cellular changes, I want to introduce the Pantom Detect test. Pantom Detect is a blood test by detection of enzymes DNase X, is APO10 and TKTL1, which occur both in elevated concentration in all cancer types. The test is based on EDIM technology for detection of special enzymes inside immune cells, macrophages. The importance of TKTL1 as a trigger of malignant transformation has been elaborated in recent studies. The expression of TKTL1 and DNase X as its companion biomarker for proliferation can be reliably measured in macrophages. This is indicated by results of studies that have investigated the technical functionality of the EDIM technology, but also by the results obtained in studies with tumor patients. Since TKTL1 formation is also upregulated in inflammatory processes, the EDIM technology using only the marker TKTL1 runs the risk of detecting not only the presence of malignant cells, but also the presence of inflammatory foci without the possibility of discrimination. This is because the EDIM technology does not allow differentiating between TKTL1 containing macrophages from inflammatory lesions and those from tumors. This risk could be significantly reduced by a co-identifying marker that is found in tumors but has no link to inflammatory processes. For this reason, it was decided to use a second intracellular marker and to evaluate it in the context of TKTL1. This is an epitope of deoxyribonuclease, DNase X, and endonuclease involved in programmed cell death. Overexpression of DNase X indicates inhibited apoptosis and appears to be an early event during the development of precancerous lesions. DNase X does not respond to inflammation. It's a universal marker for tumor activity in general, without differentiation between benign or malignant. The joint use of the markers TKTL1 and DNase X effectively reduces the detection of inflammatory processes and provides indication that the process of malignant transformation in tumor cells is active or has already been completed when very high TKTL1 levels are detected. Together with the advantages of the EDIM technology, this results in the best possible combination for a blood test for early cancer detection so far. For the processes how to do this, I will skip here because of time. But coming to the processes itself, repeating the simple blood test every year, it improves the chance of detecting cellular changes in time. In our clinical work, we can see that in very early stages, only APO10 is elevated. Then also TKTL1 is coming up. The algorithm of both parameters characterizes the turning point. And how do we combine these biomarkers with imaging procedures to close the gap? Now, Pantom detect can provide first indication of dangerous tumor growth, but cannot provide an indication of location of this growth. If patient anamnesis and further examination do not provide clues of tumor origin, modern imaging procedures such as MRI or PET-CT are needed to confirm and localize tumor suspicion. Coincidentally, FDG PET-CT makes use of the increased energy metabolism characterized by the requirements of more glucose in cancer cells to detect cancer. And as described earlier, TKTL1 is the responsible trigger for this elevated energy metabolism in form of the pull effect described by Li. There's a direct rationale between increased concentration of TKTL1 and the accumulation of radiolabeled glucose in tumor cells, which can be visualized in PET-CT. This makes the combination of the marker TKTL1 and FDG PET-CT examinations ideal for cancer screening. The pretesting by an inexpensive and low-stress blood test enables the targeted use of FDG PET-CT for screening. The screening program is aiming at healthy people. They take a blood test once a year. Abnormal results, about 2 up to 3% of the examined subjects, are clarified with further diagnostics. Imaging procedures up to PET-CT are used. Patients diagnosed with cancer are then given specific treatment. In a study with more than 5,000 supposedly healthy subjects, imaging procedures were performed on 151 people following the positive Pantom detect result. In 124 of the 151 people, the imaging provided evidence of cancer or precancerous conditions, thus confirming the result of the Pantom detect test. This result indicate that the Pantom Detect blood test could be used as a screening tool and, in combination with PET-CT and MRI, enables the detection of malignant tumors and pre-malignant lesions at a stage where, in many cases, there's a good chance for a cure. If we do not have a specific test available for each entity of cancer, or if we have too risk for screenings and techniques, or screening of the population will be too expensive adding all specific tests, one option could be to use this metabolic approach in a combination with imaging procedures to add information for early detection of cancer. To sum up, prevention and early detection of cancer is one step to longevity. Combination of immunological biopsy and imaging technologies can help for early cancer detection and maybe can improve survival rate and also keep life quality up. New partnerships between insurance companies, as we did in Germany, industry, political opinion leaders, and physicians can shift the actual paradigm from illness to healthiness. A pilot project within an entire health system would be desirable to prove the benefit for an entire society. So thank you for your attention, and if you have any questions, don't hesitate to contact me. We are looking for partners for investigations and all other situations. Thank you for your attention and have a great afternoon.
Thank you. Questions?
I have a question if this method can be used also for hematological tumors.
So far not.
Okay, did you test it?
Yeah, we have seen no very good correlation in this. So it's good for, let's say, tumors in organs. So far, more than 40 up to 45 different tumors could be investigated by this technology. It doesn't work very good on skin cancer and blood cancer so far.
And can it be used also for pediatric tumors? I'm asking this because sometimes in pediatric tumors, let's say osteosarcoma and Ewing sarcoma, there is a problem in diagnosis and confirmation, and there are invasive procedures in order to understand, can it be used like not for like for early detection or to validation that actually there is no metastasis of Ewing sarcoma?
Well, first of all, to mention, this test is in that situation only used for healthy people, not for already persons having any oncological problem. And because it's a very new approach, we focus on people between 35 and older. Probably because the technique should investigate in all ages, I would guess that it should work, but more research is necessary for that.
Thank you.
There are some researchers that claim that there are many small tumors in people that would not develop into the deadly ones, and wouldn't you, with this type of screening, have the effect of a lot of false positives, but really a lot of false positives, because the tumors do exist, but they would not develop into something. I mean, I read that someone that analyzed women that died in circulation accidents. They were about the age of 50, and half of them had breast cancer, small breast cancer tumors, half of them.
I don't think this, because it's a combination of two markers. APO10 is a marker for, let's say, a tumor, benign or malignant. But changes in TKTL1 is a marker for changing metabolic situation in the tissue and in the tumor itself. And that is the, let's say, there is a critical turning point when TKTL1 is getting higher and higher, where there is, let's say, it's a one-way situation. And that makes, I think, the test also very interesting, because you can differentiate between the beginning tumor and seeing, let's say, at the beginning, small amounts of APO10, maybe very, very small amounts of TKTL1. But there's a specific algorithm used to identify the turning point from, let's say, tumor to cancer.
Hey, is there any connection between this gene and cancer in Neanderthalim? How do you say it in English?
Neanderthal.
Yes.
Yes, there was a research done that this enzyme makes it possible to overcome the status of Neanderthal and going to the Homo sapiens. This is a different story, a long story, and probably Dr. Johannes Koy could explain it much better than me. But indeed, there is a link between. It's that enzyme which makes the change.
So it means they have less cancer?
No, I wouldn't say it's a different metabolic situation, and it protects because it's not a pro-oxygenic pathway. It protects some tissues. And for example, in our retina, this lactatic pathway is more prominent and more necessary, because if we would have too much free radicals, we would destroy retinal function, and that makes the TK-TL1 enzyme and pathway so interesting for the further development of Homo sapiens.
Okay, thank you, Maria, for your, I would say, very promising presentation. And I wish you all the success in your future work. Thank you very much.
Thank you.
Thank you. Now we are moving to the next speaker, Dr. Arseniy Trukhanov will talk about the handbook on healthy longevity, the roadmap. Please.
Dear colleagues and friends, on behalf of European Society of Preventive, Regenerative and Anti-Aging Medicine, I'd like to congratulate our friend Ilya Stambler and his team with this great event, scientific event, Longevity Nation Congress. And we are participating for the second time in this Congress. From European Society, we have 5 members of board who are participating in this event. Professor Statis Gonas, the chairman of our scientific society, who presented his lecture yesterday, Professor Marius Kyriazis, Dr. Mario Krause, Ilya Stambler, who is a member of our board, and me. Would like to share our experience and our thoughts on what we consider as longevity medicine and what is the principles of the clinic for longevity medicine and some of these methods and definitions. First of all, we would like to consider that the basic methodological document is the ICF, International Classification of Functioning, which was approved by World Health Organization in 2001, and it includes diagnostic evaluation and evaluation tools in the field of physical and rehabilitation medicine. Also, what is important for us, it's documents of United Nations Sustainable Development Goals, which was approved for the first time in 2015, and followed by World Health Organization Global Strategy and Action Plan on Aging and Health. And the strategic objective 5 in this document is stating that our goal is improving measurement, monitoring, and research on healthy aging. And due to the concept which was approved by European Society of Anti-Aging Medicine, we have several decades. In 2002, we had only clinical medicine, and we followed detection of the problems in the organism and were looking for the organ which is responsible for this problem. But then in 2003, we got revolution in the medicine with getting the first full sequence of genome. And with this revolution, we planned that in 2022, we will arrive at molecular medicine or biomedicine. And at this level, we will not only consider organ and its problem, but also we will look for the cell, genes, and the environment, and comes to arrive at the new story, which we call epigenetics. And the longevity medicine is the future of our roadmap. We consider that we will arrive at this step in 2032 when we can treat our problems, future disease on the cell and gene level using gene engineering and gene therapy. Based on our experience for the first 20 years of our roadmap, in 2022, we published our first volume, which was called Handbook on Anti-Aging Medicine, where more than 20 experts from Europe participated, and this book was had included 4 main parts: theoretical aspects of anti-aging medicine, advances in regenerative and aesthetic medicine, and personalized preventive medicine, and methods and technologies for personalized interventions. And what is longevity medicine methodology for us today? First of all, we should present you this definition, how we consider longevity medicine. It's all medical methods that are aimed at delaying, stopping, or reversing age-related process in the body. It's a holistic approach that leads to increased vitality, improved quality of life, and healthy longevity. Then last year on this Congress, Longevity Nation, we discussed these items with Ilya Stambler, and normally Ilya initiated the second volume of our YESAM library, and the concept of the book was to consider what is longevity medicine. Is this a result of the development, wellness and medical spa center, anti-aging practice, or clinical medicine? What is the contribution of academic medicine to the future of longevity medicine? What is the role of anti-aging practice like 4P medicine in the longevity medicine of the future? What are the protocols of regeneration and restoration technologies for the longevity medicine? And what are the pathways and crossroads to health longevity? And the main goal of the project was to make our vision on the philosophy, structure, methods and technologies, practical protocols, and guidelines for the physicians of the future. Finally, we arrived at the content of the book, which included 3 parts. This time it was clinical aspects of longevity medicine, part 2, components of success of the longevity clinic, and part 3, the concept on longevity medicine development. In the first part, we would like to consider the immune aging, and this chapter was presented by Professor Krause from Italy and his team. And he stated that decline in immune function with age presents significant challenges for the health and well-being of all the adults. Also, the reduction in the number of blood naive cells, coupled with an increase in the frequency of memory cells and the presence of inflammation, are considered key hallmarks of immune ascent. And he presented some of his experience with age-associated changes in natural immune cells. And here you can see a chart with a cell, a type of the cells, neutrophils, monocytes, dendritic cells, natural killers, the phenotype and changes which occur with aging. Also, we presented these schematic changes which occur during immune aging with people. Is thymus involution, overall changes, K lymphocyte changes, B lymphocytes, and inflammation process. Also, interaction of sex and gender in defining immune aging through immunobiography assessment was considered. And here are some thoughts from Krause and also our partner, Professor Claudio Francesca, who was the founder of this theory, immune inflammation. And the second theoretical aspects, which we considered in the first part, was neurodegenerative disorders associated with the 3 radical damage. It was presented by the team of Professor Vittorio Calabrese, current president of our society, And he made a statement that several neurodegenerative diseases like Alzheimer's disease, Parkinson's, multiple sclerosis, Down syndrome, ischemia are associated with disorders and with free radical damage. And in this chapter, we presented different hermetic nutrients which can play a role in healthy aging and longevity. And Professor Vittorio Calabrese confirmed that the balance has become the main subject of intense study, particularly those focused on brain aging and neurodegenerative disorders. And this process is accomplished by a complex network of the so-called longevity assurance process, which are composed of several genes termed by Vittorio Calabrese like Vita genes. Here we can see some chemical structure of flavonoid and relative components like flavanol, anthocyanin, flavan, curcumin, chlorogenic acid, which play important role as anti-aging or longevity nutrients and can be used for protection against future neurodegenerative disease. In part two, we consider the experience of the Wellness Academy group headed by our ESAM Asia director, Professor Mike Chan from Malaysia, who shared his experience in creating wellness clinics. And the main approach in wellness clinics is to restore the natural structure and functions of the tissues, organs, and cells by removing harmful factors, external or internal. They have their own proprietary health management concept based on diagnosis, detox, repair, and rejuvenate. And diagnosis include genetic screening, body composition, biological age study, physiological tests, stress detection, and different other items. Also, detox is the next important step in regenerative treatment. And the repair protocol includes intravenous infusions of vitamins, activators of mitochondrial oxidation, and precursors of the neurotransmitters. And rejuvenate methods are based on stem cell peptides for anti-aging therapy. Based on our European experience, we consider the following structure of longevity clinic in the future, which will include, on one side, traditional departments like medical fitness, medical spa, wellness and relax zone, detox and infusion therapy, also Department of Regenerative Medicine, but first of all, it's preventive department, which will include preventive cardiology specialist, preventive neurology, preventive oncology, which we mentioned in the previous lecture of Dr. Mario Krause, dermatology, endocrinology, ophthalmology, and dentistry. All these specialties which define, on one side, prevention, but on the other side, the quality of life of our future healthy patients. And the part three is the main part of our colleague today, who first of all presented us the longevity ecosystem in Israel with many official bodies like Ministry of Health, Ministry of Science or Technology, the Planning and Budgeting Committee of the Council for Higher Education, the Israel Innovation Authority, Israel Science Foundation, and others who are participating in the Israel ecosystem, which may be an example for other countries to follow. And finally, last month, we arrived with the handbook on longevity medicine, the roadmap. This roadmap we planned for 2030, and now you can find this book in social medias, in internet. And I'm very proud that the team with more than 25 specialists from European countries, from North America, from Asia participated in this book. And the majority of these offers were presented during these days of the Longevity National Congress. Here are the members of our board of European Society of Anti-Aging Medicine, and it's the Clinic Real Life Active Longevity Institute, where I'm now managing as a scientific director and implementing all the methods, technologies, and the principles which I presented in my short presentation. Thank you very much for your attention.
Thank you. Any questions? What is the price of the book?
The price of the book is nearly $60.
Oh, okay.
Yes, it's about 300 pages. It's about 300 pages, 315 pages.
Okay. Thank you, Arseniy.
Thank you. Thank you very much.
Okay. Now we'll talk my co-chair, Yaky Yanay. We will wait if Enrica Mairo will appear or not. It's under the question. So, please, from the best to renewal, the therapeutic potential of placental cells to promote healthy longevity.
Hello everyone, so my name is Yaky Yanay, I'm the CEO of PluriBio. I'll keep it short and sweet, talking about how we're using cells in order to promote well-being, aging, and longevity. And I did tell it to Ilya privately during lunch. I applaud for you making sure that this conference will happen, no matter the situation he actually is, and you have my full appreciation for that. So thank you, and for all the great work that you are doing all over the years. So well, guys, that's a problem. That's the source of the problem that we are fighting today in our conference. Why are we dying? And I don't know if you made it by purpose, Ilya. Surprisingly, that was actually the Parashat HaShavua. That was the chapter of this week in the Bible that we read every year, every Shabbat, the chapter. It's actually the Bereshit. And guys, that's probably, according to Jews, the reason that we are dying, and this is the reason that we are aging. So that's the source. And everything that we are doing since then is to try to find solutions to what Eve or Chava was doing, eating from the fruit. And we are somehow trying to see how we can keep ourselves alive, even though this is how we started. But talking about Eve and women, we are the placenta guys. I love placentas. I'm amazed by cells. And we speak about it for years, but in my opinion, it just makes sense to take young, potent cells that are coming after full-term delivery and using them in order to regenerate and to heal our bodies. Generally, cells, in my opinion, holds a huge promise to many of humanity challenges and that we are facing. Because everything on this planet that is living is built from cells. I like to call cells the building blocks of life. We are built from cells, our food, trees, the tiniest bacteria. And my goal in life, and this is what my company is doing, is trying to create product out of cells. And for therapeutic, I believe in placenta. I believe it's quite an amazing organ, very interesting from immunological point of view. And we can use young, donated placenta in order to create therapeutics. In Pluri, we invested quite a lot in building and technology that can allow us to manufacture cells on scale. And we invested heavily, over $300 million. What allows us today to have this bioreactor system that we can expand cells in a dramatic manner. Today, we use it across different industries. But these systems are so effective that from a single placenta today we know to manufacture over 20,000 doses. So we can treat over 20,000 patients from a single placenta. We do work with several types of different cells that are deriving from placenta. And what we know that once we are administering these cells into the patient body, the cells starting to interact with the patient body. They are able to get signals from the body and to respond to it by secreting a variety of different proteins, cytokines, chemokines, exosomes, which eventually leads to regeneration and healing. The advantage of using cells, it's a living organism. And for me, it just makes sense to treat a body which is built from cells with cells. I always say placenta cells are native speakers. They understand the language of the body. So they can read the signals and respond to it. And this is what we are trying to do in all the medicine all over the years. And this is what Pluri is expert about. We work for 15 years using one type of cells, the adherent stromal cells derived from placenta. We have several products. We use it quite successfully, treated hundreds of patients from ischemic condition, hematological deficiencies, muscle regeneration, and more and more and more. Today we are also using a different type, which is a non-conventional T cell derived from placenta in an immunotherapy fashion in order to combat cancer. And I like the title of the presentation of if you like to stay alive, make sure that you're not going to get cancer. It's true. But if you're getting cancer, so we need to treat it somehow. So we are all about placenta. It's about different cells and about using these cells for therapeutics. But that's a problem, that we use cells for therapeutics and not for well-fit, healthy human beings. And this is the big shift that all of us want to do at a certain point, how we can do prevention. And if we are, as human beings which are in good health, are eligible for cell therapy treatment in order to keep and to preserve our well-being, we are talking, for years, over a decade, I do try to understand the aging phenomena, and we all know it very well. Complex indication or complex conditions that are coming from different organs, from ischemia, muscle wasting, the age of our immune system, the fact that our cells get into a senescence mode, inflammation. It's a real complicated and comprehensive solution that we need to overcome of it. What is interesting, that if I'm going to slice and dice it per indication, today I have good clinical evidence that we can use these placenta cells to treat any of these conditions separately. Why separately? Because today, unfortunately, I'm treating patients. I hope that the day will come that we're going to treat healthy human beings in order to make sure that we're going to keep our well-being and aging and longevity. And let me give you a few examples to understand what we can do with placenta cells. One of the most important parts, and we all know, is the muscle wasting. We can easily see aged people by their skeletal muscle fitness and volume. In Pluri, we did several studies with the Charité of Berlin, which is our European partner, but in a quite large phase 3 study. We were able to demonstrate that if we inject these placenta cells to cells into muscle, we can increase the muscle volume and we can increase the muscle force in a statistically significant manner. This is some finding from our phase 1 and phase 2, but we also repeated and published the phase 3 study that we measured. So once the cells are injected into the muscle, they interact, they secrete a lot of factors that allow the muscle a much better and faster regeneration, and we get a stronger and a larger muscle. Well, that's great for patients after hip fracture, but why not to use patients or healthy people that are just aging and from 25 losing the muscle mass? I think we should do it. Ischemia. We know that ischemic condition is a part of the aging. We see ischemia in the leg, in our hearts, in all our arteries and blood vessels. We have a good evidence that if we are treating patients with a severe ischemia, in this case, again, end-stage patient with a CLI patients that are in a risk for amputation, we are able to save the legs because our cells secrete factors that allow the body to start and to create additional or new blood vessel by doing angiogenesis, the natural angiogenesis that the body know how to do. So it's all about it. Our body know how to regenerate. We are losing it at a certain time, or it's slowing down at a certain point of time when we are aging. And I believe that it's as simple as that. Take young, fresh, potent cells, give them to aged human being, and you can see some interesting stuff going on. I guess most of you know about a lot of studies that was done on mice, seeing that if you take young blood and inject it to elderly mice, you can see the outcome. So our cells, and we have a different type of cells that we're using for different disorders. Pluri always work in collaboration. This is a great collaboration that can give us also some indication for the hematological deficiency. We know that the age of the immune system is a critical part of how well we are aging. We have quite an interesting program today that we are doing with the Department of Defense and the NIH, using our cells to treat hematological deficiencies after exposure to very high level of nuclear radiation. And again, I'm treating patients. I want to treat healthy human beings, but we know that our cells can be very impactable over there. And for the cancer itself, we are developing now, it's relatively an early stage, but quite promising, an immunotherapy model based on placental cells in an allogeneic fashion. So we hope and believe that we can overcome the shortage of the supply of immunotherapy, which we all know that it's effective, but still very costly and with quite limited access to patients. And that's something that I hope that next year we will be able to report on more success on that. But working with placenta cells, expanding them, using the immunotherapy, that's something which can be a very powerful component. It's not relating to aging, but just to tell you what I have in my mind. I believe in cells. Cells are important for health, but they're important for many, many more different industries. And I urge any one of you that have a good idea to contact me, because in Pluri, we are actually building a collaborative network of companies. We are building different companies around us that we are spinning them out, always using the same platform that we have. And in the last 2 years, I did things that I never thought I'm going to do, like building a company with Nuva for cultivated meat, using our technology, which was for expansion of placenta cells, but now for meat. We are now partnering also around AgTech for several applications, always the same thing. If you have a great idea that you need the most amazing manufacturing technology of cells in the world, we are building a series of companies around us. And the company that I'm looking forward to see it very soon is around aging and longevity, that will devote all its energy towards building solutions for aging and longevity. It needs to be a separate one from the pharma because it's not treating patients. And I started with the Bible, and I will end with the Bible. One of the promises, or one thing that we know that we also read it this week, and I always say, the first man Adam, he lived to 930 years. So who knows? Maybe this is our biological potential. We need to do a lot of good work. I can say that I hope that next year, Ilya, we're going to see significant progress in aging all over the world. I think that all of us did an okay work, but we can do more than that. And this is where we should look for it. The first man Adam, 930 years. Here's the goal. Thank you.
Questions? I understand that you generated industrial processes for generating multiple cells, basically. In this case, placenta cells. So can you use the same processes for personalized medicine? I mean, taking a cell from someone and multiplying for the purpose of later turning them back. I mean, isn't it more or less the same industrial process for that?
I think it's a different manner of doing things. To do it in a personal manner, you actually don't need our technology because you can do it on a small scale. In Pluri, we think on billions, hundreds of billions, trillions of cells, because in my opinion, cells should be the first line treatment. Before going to chemicals or antibodies or whatnot, Try to use cells. They are safe, they are good, and they are potent. Can we expand different cells? Absolutely. From iPSCs to T cells to, of course, MSCs, placenta, bone marrow, fat, everything that look like cells, talk like cells, and walk like cells, we can do it. And also cell and cell extract, including exosomes, mitochondria, and more and more and more. It's a really strong platform that we can work on it. For personal treatment, it's overkill. We are looking on mass production.
Hi, thank you. And I would like to ask, when you inject the cells, how many cells do you inject, and how long do they engraft in the body? Do they die? Do they disappear? And how do you think that they, for example, alleviate bone marrow injury when you inject them?
Cells, and especially allogeneic cells, it's of course, we spent a lot of time to understand the mechanism and how long they stay. By being allogeneic, they do not engraft. So they stay for a while in the patient body. Our studies indicate that we see them 3 even to 6 months after injection, but the number is going down over time. The number of cells that we're using depend on the indication. What we did learn that on acute conditions, like hip fracture, for example, that we need a short-term regeneration, you don't need so many cells. We did a single administration of 150 million cells, and we got a very good outcome. On a chronic conditions, like ischemic leg, which is actually a full-blown systemic disease, we are doing a higher dose, a dose of 300 million cells, and we are doing repeat dosing because you need to continue to do the regeneration. The body is not in a great condition. So independent of the indication itself to do it. In that extent, placenta cells are quite unique, and this is why I love placenta so much. It allows us, even though they are allogeneic, they have a very low immunological presence in the body because they're coming from pregnancy. And pregnancy is the only situation in nature that I know that you have 2 immune systems that coexist, the mother and the baby. And we know that the mother can be even a surrogate mother. She's going to carry someone else's child. So placenta cells are quite interesting from an immunological point of view. Very low HLA markers, which allow them to stay for long and not to create any of immune response of the body against them. Not immediate immune response, but they don't graft, they're not part of the body.
I have a question about, do you really need cells, or you need what cells are actually producing, all the secretome, exosomes? Did you try without cells, like just, because sometimes cells can make problems in the body, right? Like if you want all the goodies from the cells, why not to do the same with molecules that can do the same thing?
Yeah, it's a fair question. Secretome, and nowadays it's quite popular. Everybody talks about exosomes and secretome and more and more and more. We're doing it. And today we can use it. Well, I'm maybe kind of a romantic guy, but my clinical evidence always tells me that if you're using the cell as a whole, you get a much better outcome. So it's not only about the secretome that now is in the cell itself. These guys are responding to the environment all the time. So if they are sensing, for example, an inflammatory environment, they will significantly elevate the anti-inflammatory secretion. And you're having like a pump that has a relatively long half-life time, and it's responding to the environment. And this is, in my opinion, the secret or the most important part of cells. I call them a drug with intelligence. Because of the ability to interact and communicate, on a secretome level, you lose it. You have what you have, and sometimes it's great because it prevents some of the complication of using cells, but you get partial part of the power of cells, in my opinion.
Do they make problems sometimes, causing cancers or so on? Are any evidence on that?
So as of today, we've treated, it would be fair to say, close to 1,000 patients all over the world with a very, very good safety profile. The one problem with cells that they never get toxic. So we're seeing in preclinical studies that we injected unbelievable amount of cells. Because they are biological material, at least these mesenchymal cells or adherent cells, they are very, very safe in nature. But you're losing potency if you inject too much. And it was part of the lecture before that we just said that we're using the same frame. Sometimes too much from a good thing, it's not good. You're losing part of the potency. This is what we see. You need to find the right dose to keep the efficacy, but on a safety, it's very safe cells.
Thank you.
Thank you.
See how a meeting gets more and more optimistic. Thank you.
I hope so.
Yes, for your presentation.
Thank you very much. So we're going to go for the next presenter, Professor Robert Chunazaho from the School of Basic Medicine in Peking Union Medical College, the application of MSCs against aging and related disease. He's going to present.
Just a small correction. It's the same Peking Union Medical School and the same presentation, but the presenter will be Dr. Qian Li from Chunhua Zhao's team. So you know.
Thank you. Please.
Good afternoon, everyone. It's a great honor for me to have this occasion to present our work on behalf of Professor Chunhua Zhao and our team, focusing on the application of post-embryonic pluripotent stem cells for the treatment of major diseases, including those related to aging. As other speakers have highlighted, in treating complex diseases, we have entered the era of cellular medicine. Among all cell-based products, stem cells are particularly valuable due to their potential for tissue repair and regeneration, immune modulation, inflammation reduction, and oxidative stress mitigation. These are actually the key mechanisms that make them promising candidates for anti-aging and other therapeutic applications. And this has driven a global surge in stem cell development over the past decade. In the early stages of developing stem cell technologies and therapies, we faced major challenges, from sourcing quality stem cells to advancing key engineering techniques and ensuring the safety and efficacy of stem cell products. For the first time in the world, our group leader, Professor Zhao, leading our team, identified a subpopulation of stem cells that exist after the embryonic stage of development. We term them as post-embryonic pluripotent stem cells, PPSC. Since PPSC have the capacity of differentiating into all cell types within the ectoderm, mesoderm, and endoderm, we propose they are the ideal seed cells for cellular medicine, engineering, and industrialization. We characterized the biological properties of PPSC at both genetic and epigenetic levels. Key phenotypes, including positive expression of CD29 and negative lean expression, support their stemness and multi-lineage potential. Importantly, PPSC lack expression of OCT4, have weak non-OC expression, and exhibit high KLF4 levels. This gene profile mitigates tumorigenic risk while retaining sufficient stemness, supporting both the safety and differentiation capacity of these cells. Upon further exploration, we discovered that PPSC exert both anti-inflammatory and pro-inflammatory effects. For instance, PPSC can inhibit the differentiation and maturation of dendritic cells, while also promoting their function by inducing the generation of a subpopulation of regulatory dendritic cells. This important characteristic enables PPSC to maintain tissue and cell homeostasis. So we propose that these bidirectional immunoregulatory effects serve as a theoretical basis for the effectiveness of PPSC in treating immune disorders. These findings are listed in the faculty 1000 database and have received widespread acknowledgement and positive feedback from internationally recognized specialists and institutions. In the special report in immunity, the discovery of PPSC was highlighted as offering promising therapies for major diseases, including organ rejection and autoimmune disorders. Additionally, our research results have garnered worldwide attention and have been cited in numerous peer-reviewed publications by esteemed figures. For the industrialization of PPSC, our R&D team established China's first industry standard and the core system for the large-scale application of adult stem cells, accompanied by a series of patented technology standards. Due to our identification of PPSC and the corresponding drug production standards, we have received numerous awards and have been recognized as an industry leader in adult stem cell engineering. In 2004, we obtained China's first approval for a stem cell clinical trial aimed at treating leukemia, followed by another approval in 2006. Our long-term clinical studies demonstrated the safety and stability of PPSC, with the overall treatment efficacy for leukemia reaching up to 88.9%. PPSC-derived drugs have also demonstrated significant potential in treating severe cases of graft versus host disease, GVHD, particularly those resistant to traditional hormone therapies. Due to these positive effects, we received the first approval for a phase 3 clinical trial of stem cell drugs from the FDA in China. Over the past 16 years, these trials, conducted at multiple national hospitals, have confirmed the safety and effectiveness of our stem cell drugs in treating GVHD, achieving a treatment efficacy of 78%. As our indications expand, all PPSC-based pharmaceuticals undergo rigorous and repeated evaluation for clinical effectiveness. For example, the stem cell drug for blood disorders has been clinically validated for 15 years, while the drug for heart diseases has successfully treated over 100 cases. Long-term efficacy and standardized research protocols remain our primary evaluation criteria. Based on our PPSC technology and promising clinical results, we successfully developed China's first, second, and several other stem cell drugs targeting severe and refractory diseases. These drugs have been shown to be effective in preclinical studies and are currently undergoing extensive evaluations across all stages of clinical trials. In response to the global public health pandemic in 2019, particularly considering its disproportionate impact on aging populations, we have been leading key projects to investigate the potential of using PPSC to reduce the severity of COVID-19 and its related complications. In a pilot clinical study, we showed that MSC, mesenchymal stem cells, prepared through our PPSC system significantly alleviated COVID-19 symptoms, reduced hospital stay durations, and increased cumulative remission rates. The therapeutic mechanism involves the suppression of overactivated immune cells and regulation of inflammatory cytokine release. Importantly, PPSC-based MSCs exhibit minimal ACE2 and TMPRSS2 expression, reducing their susceptibility to coronavirus infection, and their high expression of multiple growth factors underscored their strong immunoregulatory capabilities. Following our pilot study, we conducted a randomized, single-blind, placebo-controlled phase 2 clinical trial to further assess the safety and efficacy of ACE2 negative MSCs in treating patients with severe COVID-19. In the peripheral blood of patients treated with MSC infusion, we identified a unique group of cells expressing VNN2, a marker associated with immune and hematopoietic processes. This finding suggested the mobilization of hematopoietic stem progenitor cells from the bone marrow in response to MSC treatment. Further analysis revealed that this key subpopulation of HSPS, PC, expressed 171 genes linked to angiopoiesis, HSC mobilization, and fetal extramedullary hematopoiesis. Such extramedullary hematopoiesis may support immune function activation, suggesting that this bone marrow-derived cell population could play a crucial role in sustaining immune responses against coronavirus infection. Further supporting this finding, we find PPSC-based MSC helped restore immune homeostasis and promote recovery in COVID-19 patients by modulating various immune cells. They do so through upregulation of chemotaxis-reduced genes in different cell populations, promoting T cell differentiation into helper cells, inhibiting overactivated immune cells, activating B cells to support tissue repair, and suppressing neutrophil extracellular trap formation to reduce the risk of immunothrombosis. To summarize, the molecular mechanism of PPSC-based drugs in improving COVID-19 prognosis involves multiple actions, including combating viral infection, promoting tissue repair, and protecting the body against the severe complications such as thrombosis. Our reports on stem cell treatments for COVID-19 were widely recognized by the international community and hold significant clinical application value. Notably, our phase 2 clinical trial was listed as the first reference in the US NIH guidelines for COVID-19 treatment. As a testament to our efforts in utilizing PPSC theory and the system to treat major diseases, Professor Zhao has been recognized as one of Elsevier's most cited scholars in China for 10 consecutive years and was named among the top 2% of the world's top scientists in both 2022 and 2023. Now let's take a moment to review our work on stem cell development and therapies. To date, we have validated the hypothesis and system of PPSC, identified an optimal stem cell seed with immunomodulatory functions, achieved large-scale production of adult stem cells with strong tissue repair capabilities, and established evaluation protocols for clinical safety and feasibility. We feel that our research reflects a commitment to integrity, from the development of theory to the advancement of technology and products, and from those products to industrialization and clinical application. We are grateful for the recognition and support we have received. So finally, I would like to express our heartfelt gratitude to the funding agencies for their support. Thanks for your attention.
Questions? Comments? I will say one thing, and that's thanks to Ilya. During COVID, you do connect me with Professor Zhao, and that's eventually we can actually confirm your finding. We did a parallel 80-patient study in the US for ARDS COVID patients, and Professor Zhao initial findings were led also to these studies showing a significant increase in survival using these cells. So thank you for making all this contact. It's not about aging, it's more than that. Any more comments? Thank you very much. And now we're going to move to Professor Kunlin Jin from the University of North Texas, Health Sciences Center and International Society of Aging and Disease. The key players of the pathophysiology of age-related brain disease. Thank you very much. Professor Kunlin, please.
Thank you for invitation. After several years of clinical, practice, I realized that, the people die not just purely aging. People die because aging-related disease, like cancer, cardiovascular disease, stroke, and AD. So we are not just want to stay longer, we want to stay healthy. This is why my talk is not just focus on aging, but also focus on the aging-related disease. As early in the 18th century, the life expectancy roughly is 37. In the early the United States, the life expectancy roughly 47. Now in 2022, roughly lifespan in the United States is 77 and 78. The currently, the longest, the life expectancy in the global roughly mostly is Hong Kong is 84.5. I think last year, this year I went to Hong Kong, they say 87 now. Then Japan, Singapore, and Australia. So you feel that it's most of those countries near the ocean. Actually, it's maybe not the link to the seafood. For example, in Hong Kong, the people living longer because the health insurance, medication, most of the advanced medical equipment and medical, the drug like medication. So people can, as long as have the early sign of the disease, they can get the treatment. So the lot of the debate regarding why we get in aging. So one of the lot of theories like a program theory and free radical theory and DNA damage theory. So none of them can explain the well to why we get in aging. So I think that one of the major the theory is microcirculation impairment. This is the many cause of the aging. The main function of the microcirculation is deliver the oxygen and the nutrition and remove some toxicity and also the CO2 from our body. Once we microcirculation impairment or dysfunction over the time can result in accumulation of the toxicity products and the CO2 in our body and the loss of the nutrition supplement and oxygen in our corresponding tissue, eventually affect normal, the organ function or cell function leading to the aging. So what's the evidence, The first evidence, the aging related with the capillary loss. So this is the brain of the animal is young rats and aging rats. As you can see that in the cortical view, a lot of the blood cell, particularly in the capillary, in the cortex, but when getting older, the number of the capillary is significantly reduced. We not just reduce the number of the capillary, but also when we're getting older, the wall of the capillary or the artery is getting thick. So this is getting thick. So sometimes it's blood cell or immune cell sometimes is difficult to diffuse, in and out from the organ. So the vascular loss is a permanent hallmark of the agent. This is the immunostaining, use the full color of the staining, indicating that spleen, kidney, there is a loss of the number of the capillary. This is the immunostain indicating that in the human section and also in the animal section, kidney, muscle, and the spleen, indicating the number of the blood vessel, particularly like a capillary and the city, small artery, you can label the alpha small muscle unit. As we can see that the number of the capillary cell, like endothelial cell, is significantly reduced when we get in the older. Another concern is that evidence is the oxygen delivered to our brain, to our body, is reduced with increased aging. There's function MRI indicating that oxygen is reduced when we get in the older. There's a cartoon showing that compared to 20 years old and 80 years old, the cerebral blood flow is reduced with increased aging. Is green matter? And the artery changes at time in the blood vessel. Also, the cerebral blood flow is reduced and ATTs increase when getting older. One of the mystery, the stuff is that the longevity in the Salento is the South Sea Island in the Italy. The average life expectancy in the Salento is right at 92 years old for women and 85 for men. In 2016, Dr. Salvatore, did some study. They have found that the blood flow in their muscle is much the stronger compared the other city. They have found that so they're in the serum or in the blood, there's one protein called BDIFB4. There, when isolate this protein injection to the animal, they increased the blood function and increase the the microcirculation. This further confirmed the microcirculation is critical for our aging process. So the microcirculation dysfunction, I think there's the key for is linked to the other aging theory, like stem cell theory, because if you don't have the blood supply, don't have the nice environment or niche for the stem cell proliferation, migration, and the differentiation. Like a free radical, if we don't have enough blood supply, the free radical of course increase there. Same as the DM, same theory and the program theory is kind of key link there. So we know that the capillary may contain the endothelial cell. Now we found that not just endothelial cell, we have also another cell called a parasite. Parasite is also looked on the capillary. There's the EM study. There's the indicating the parasite. The beauty is the parasite not like an endothelial cell. The parasite have some construction function because they contain small alpha muscle cell, the protein. So it can bind directly and control the blood flow in the capillary. This is the evidence indicating that, there's parasite construction. So that's the exo-wave study. In the middle is a red blood cell. They can pass the blood cell. When get it stimulated, parasite can construction. So it's leading to the diameter of the capillary getting smaller. So blood cell cannot pass the capillary. The parasite also, regulated by some, the glutamate and other hormone. So when we're getting older, so parasite will be consistently, contraction. So leading this even we see the blood flow in the main artery or main vein, but in some bed of the capillary because of construction of the parasite. So this area don't have the blood supply. With increased aging, the number of the parasite also decrease. There's the immunostain indicating the loss of the parasite in the aging the animal. The kidney, muscle, spleen, and the thermus. This is published in Science Advance in 2021. If parasite depletion also induces vascular regression in the region of the spinal ganglia, neuron, and in the adult mouse. See compare these slides indicating without the parasite, depletion of the parasite depletion, you see the number of the blood, the capillary also is reduced. There's control, there's parasite depletion, that's indicating that the number of the capillary also is reduced. That's published in the 2003 eLife. Transmission bring capillary perfusion and a change in cerebral blood flow in the parasite deficiency animal, the mouse. Is this the white animal mouse? This is the heterozygote, and this is the homozygote. When parasite completely depletion, the perfusion or cerebral blood flow also significantly reduced. This is one of the possibility is the number of the capillaries also reduced. So parasite not just involve the aging, but also involve the aging related disease. This is the example. Parasite dysfunction may trigger the impaired microcirculation in the Alzheimer's disease. So this is the cartoon indicating the non-A-beta depose. With A-beta treatment, the diameter of the capillary is significantly reduced. This is the left panel indicating immunostain. So we can use the mark like a BDGFR beta indicating the marker of the parasite. So on the right bottom indicating that with add A beta, without A beta, the diameter of the capillary is significantly reduced. There's an X-ray study indicating that it's a parasite truly in a contribution to microcirculation in the AD brain. This is a summary, indicating that the parasite function in healthy brain in AD brain. So parasite is not just involved like the blood flow, also involved because this is the part of the vascular units. So it also, it's the BBB, the function also is critical. This is an example, not just like AD, but also involve the ischemia stroke. This is the non-ischemia brain, the blood capillary like this, of the ischemia brain because of the parasite contraction. So this blood flow cannot pass through. This is like you can the red arrow indicating that it's discontinued. DRC imaging. This is the blood, the dots indicating red blood cell, and of the ischemia because of parasite contraction. So some blood cell cannot pass through the capillary. We are in the clinical setting. There's some ischemia stroke. We can treatment by the TPA. The TPA is an enzyme can dissolve the clot, right? So even in some patients of the ischemia stroke, even after the treatment with the TPA, the motor function still recovering. So there's a hypothesis that even the main artery is perfused, because however, the parasite in the capillary still consistently, the contraction, leading the low perfusion, and motor dysfunction. So how can we improve the microcirculation? Improve the healthy and still the longer and still healthy. So that some of the exercise, the people study that this also published last year that if you do some exercise, also revised aging-induced deep dependent decline in the cerebral microcirculation. So young age and aging with exercise. If you do more exercise, it will be increasing microcirculation and improve your healthy. This is another, the cartoon indicating that we do some exercise, and also the model, the cognition function also significantly improve that. And also HBO, so the hypoxia and high baroque oxygen chamber. So the people can sit in there. So for a certain time, maybe like 1 hour or 45 minutes later, so they did that. Okay, so the thermal response of the T cell and also this is significantly increased by over 20% compared to control. T helper cell, the significant decrease in the number of the senescent cell of the T helper cell also is reduced. So that means the oxygen is critical for our aging, and the micro circulation dysfunction is maybe the leading of our aging.
Thank you very much, Professor Jin, for this very interesting and detailed presentation. Any questions, please?
You have mentioned behavioral, so to say, maintenance therapy by aerobic exercises, but we know that in cardiological diseases, various vascular growth factors are sometimes used after strokes or heart attacks to renew vascular genesis and angiogenesis, although these processes are dangerous to tumors. Are you proposing some controlling factors, endocrinological factors, or biofactors that could maintain slower degradation of microcapillary system? Thank you.
It's a very good question. So actually in the clinical setting, even like I have the cardiovascular disease or stroke at the acute phase, like first couple of days, we are not a recommendation for any doing some exercise. But however, after the subacute phase, so we suggestions that we do some exercise. There are two types of exercise. One is active, one is passive. If patient is still on the bed, is not able to do some exercise, the nurse or other nurse assistant can help do some the rehabilitation, for the move the leg slowly, gently. It's gradually increase the intensity of the exercise. Particularly for the stroke and the cardiovascular, disease, the cardiac, circulation is very critical. So most of the time, like heart attack or stroke, because the blood vessel have been occluded. So it's difficult getting reperfused, as this area is damaged there. But if you increase cardiac adjacent circulation, also increase angiogenesis, those is well documented that the exercise will be increase the angiogenesis and so cardiac circulation. Particularly, it's like the VEGF, the when you do some exercise, the VEGF is increased. VEGF is not critical for the angiogenesis. We study indicating that a lot of the neuron and other type of cell also expressing the VEGF receptor. So they are not just increase the angiogenesis and also have some the neuroprotection effect. So the kind of the combination and the therapy mechanism.
Hi, thank you very much for this wonderful lecture. I'm also working on capillary dysfunction in aging, and it's really nice to hear you. And I wanted to ask you if you think that the pericyte dysfunction begins with endothelial cell dysfunction that disrupt the signal between endothelial cells and pericytes, or it is the other way around?
This is a very good question. Actually, we don't know. This is a very good area to study that, but which is leading cause and but for some study that in a published in the Science, I think a couple years ago, when we get in the stroke, the parasite will be detached to capillary, so they move away. But those cells maybe they're dead. The parasite may be dead, maybe they move to different region. Is some of the components for the neurogenesis, but we don't know. That's just a hypothesis.
Yeah, thank you.
Okay, the question is, you presented sickness of vasculature with aging, but I think it is believed that with neurodegeneration there is leakness of blood vessels. So are both processes exist?
So yes, the BBB, is damaged. When BBB getting damaged, they are leakage, particularly for we call it dysfunction. So sometimes, the wall getting sick, maybe like not just a cell, they're getting the number is increased. Actually, it's not. So getting sick is the wall getting sick. It's there's some deposit of the fiber and also the lipid, and there will be increase. So the tight junction sometimes is not so tight. So that's why when you do the blood test, you can find some even like a neuron cell, H cell in the principal blood cell in the AD patient. So because there's some cell can leaking from brain through the blocking the BBB to our body. So microRNA diagnosis is also protein marker is for the diagnosis of the AD or PD or the Huntington's disease because BBB is leaking. They both exist.
Thank you very much, Professor Jin. And now we're going to go to a presentation of the co-chair of this conference, Professor Vadim Fraifeld from the Ben-Gurion University in the Negev, Rejuvenation and Longevity Introduction of Regeneration. Please.
Thank you, Yaky. First of all, my compliments to Ilya Stambler for an excellent organization of this meeting. And thanks for inviting me and opportunity to present our book written together with an old friend of mine, Khachik Muradian, from the Institute of Gerontology in Kiev. The book was published just a month ago in the framework of the series Healthy Aging and Longevity, though to be honest, I do not understand to the end what does it mean healthy aging. But never mind. Our thanks also to Suresh Ratan for his gentle support of this work. In fact, it's the first attempt to systematically analyze what rejuvenation is and its relationships with aging, longevity, origin of life, and the existence of life as is. Many ideas in this book are to a great extent hypothetical, it's naturally, but nevertheless, we believe that during the last decade, research on rejuvenation has acquired a realistic shape and become a subject of scientific analysis. It would not be an exaggeration to say that right now a new science is emerging, rejuvenology. The book consists of 12 chapters and covers a wide spectrum of issues from the evolutionary aspects of the origin of life, rejuvenation, immortality, and aging, to the evidence that biological time is reversible. Here I will mostly address the conceptual points without going into detail regarding the mechanisms of rejuvenation and the rejuvenative interventions. So let's start from the beginning. Rejuvenation is not fantasy. It's not a nice dream. It is a fundamental and immanent feature of life. The very existence of life would be impossible without rejuvenation. It means that in particular, rejuvenation appeared first and aging appeared later. And it appeared as an adaptive mechanism for elimination of defective protocells from the population. However, in multicellular organisms, aging from the useful tool for quick removal of the cellular damage turned into the most destructive factor. Then the main role of rejuvenation has become to rid off the load of aging and minimize damage transfer across generation. Whatever, aging is not inevitable. Indeed, if the rate of rejuvenation is higher than the rate of damage accumulation, the cells will not age. It's exactly what happens in protozoa and some eukaryotic organisms, but anyway, it's not fully used in most multicellular organisms. One of the maybe the most ancient rejuvenative mechanism is damage dilution due to a high replication rate and also excretion of unwanted substances through the release of extracellular vesicles. But let's relax for the moment and remember the Sir Terence Pratchett, who said, "In ancient times, cats were worshipped as gods. They have not forgotten this." If you have any doubts regarding this statement, please take a look at 2 of our faculty cats, Ginger the First and Cat Hypo. So they're very serious, as you see. But what is common between cats and cells? Once all cells were immortal, and they have not forgotten this. Moreover, mature cells remember how to come back to youth and stemness. Putting in other words, the basic element of the living system, the cell, knows how to rejuvenate, and is potentially immortal. And the important point is that activation of rejuvenation program could be activated by relatively simple means. Why do multicellular organisms consisting of potentially immortal okay, it's not okay. Consistent of potentially immortal units have surrendered to aging so easily? At the moment, we do not have an answer to this question. Yet there are a few exceptions when multicellular species show no sign of aging or only negligible senescence, with lifespan exceeding hundreds and thousands years. We called them sechel, species exceeding human longevity. By the way, in Yiddish, sechel means intelligence. Okay. The analysis revealed a number of common features that could contribute to their extreme lifespan. We should pay attention that these species, most of them at least, live in an intermittent or permanent hypoxic environment, often combined with hypercapnia, hypoxic, hypercapnic environment. They have a low metabolic rate, and maybe to a great extent it is associated with their hypoxic, hypercapnic environment. Another important point is that these species are able to preserve a pool and high functionality of stem cells, which eventually ensures a high regenerative ability. And this feature is also associated to some extent to hypoxic, hypercapnic environment. The major rejuvenative events in mammals in particular are primarily associated with gametogenesis, with zygote genome activation, and early embryo development. The third and the final rejuvenative event starts from pre-implanted blastocyst and ends at early implantation. Note, please, that hypoxic hypercapnic environment is important to embryonic stem cells and early embryogenesis in general. This is especially true for embryonic stem cells of the pre-implanted blastocysts when hypoxia hypercapnia reaches its peaks, thus minimizing oxidative damage and preventing the embryonic stem cells from premature differentiation. All in all, this creates ideal conditions for rejuvenation. A few years ago, we wrote that these short periods of embryonic stem cell life provides a unique opportunity for the last-minute correction of the cellular damage before embryonic stem cells start to differentiate. Needless to say how much it's important. the very fact that life still exists and the accumulated damage doesn't pass on from generation to generation is to a great extent granted by the last-minute correction of embryonic stem cells. And we are pleased to say that our hypothesis gained strong support from the studies of Professor Gladishev's group, who recently showed that after the last-minute correction, embryo reaches the minimal biological age, so-called ground zero. Should be stressed that rejuvenation could take place not only in early embryogenesis. Various interventions, I will not go into details now, showed the very possibility to decrease the biological age later in life as well. This simplified model illustrates the relationships between aging, anti-aging, and rejuvenation, where the water level in the bath reflects vitality or probability to stay alive. Damage make holes in the bath, water flows out, and vitality decreases. However, the organism has tools to fix this problem, at least in part. Thus, it can prevent or slow down further leakage, but per se, it cannot restore the level of the water. Vitality could be elevated only if the water tap is open. If this is aging, this is anti-aging, and as you guess, this is rejuvenation. And something really optimistic. Human knowledge and instrumental power increases exponentially, where the complexity of aging and rejuvenation problems is a constant entity. This is a good reason to believe that sooner or later human skills will be enough for resolving any biomedical problem, anti-aging and rejuvenation included. In these coordinates, there is only one unknown: when? Will it take 30 years, 50 years, 100 years or more? Maybe it occurred yesterday. You or we just missed the latest news. Welcome, rejuvenation. Thank you for your attention.
Thank you for your book. I read it today. I think it's a minor revolutionary, very impressive.
Thank you very much.
I have a very egoistic interest in this, considering my organism. So, since you start with very fundamental things, which are microscopic, I'm a physicist. And in physics, there is a divide between microscopic effects and macroscopic effects. They are not completely reducible one to the other. How do you see the bridge between the small life of cells and you make a very strong emphasis on adult stem cells in your book as a key part of microscopy of our body, bridge to the fact that we are actually also built from macroscopic systems, muscular system, cardiovascular system, brain and neurological system. This is a little bit lacking, the bridge between microscopy and our organs. And all the organisms that you mentioned, with the exception with the Greenlandic shark and some kind of whale, their organs are relatively simple. How do you see in your imagination or your intuition, how can it be bridged to regulate that our organs will less deteriorate? Because we're not built of cells. After all, we're macroscopic systems. That's my question.
I think you answer your question. I mean the following. Sure, it will be the best way to rejuvenate the whole organism. But maybe we are not clever enough to do it at the moment. But what we can do, rejuvenate step by step, say, maybe specific organs, maybe the organ that need the most treatment or something like this. And there are some findings, observations that it could be done. You know, there are, say, interventions that can improve the liver function, and the treatment actually is based on that ancient rejuvenation mechanism of dilution the damage. As Conboy and colleagues showed, only a single exchange of replace of blood plasma by saline and albumin can significantly improve the liver function, actually making it younger. So if we are talking about a near future, it may be a rejuvenation or at least improvement, critical improvement of functions and morphology of certain organs. I mean, not the whole body, but if we will be lucky, maybe also the whole. And I didn't actually, almost, I didn't talk about stem cells. In fact, in mammals, it could be of special importance, and the really important point is that we have pluripotent adult stem cells in our body, so-called Muse cells, discovered by Marie de Zava. And another way is cell reprogramming. Cell reprogramming not only by Yamanaka factors, it may bring a lot of problems, but maybe more by using the so-called small molecules, defined small molecules that can work actually as transcription factors and make induced pluripotency. Moreover, we don't need to get this stage, induced pluripotency. Even 1-2 stage toward stemness will be also good for renewal the tissues and so on. The only, not so good news, this book, the price of this book is much higher than the price of that handbook of longevity, almost twice.
Where can we buy it?
No, Amazon, you will get a rebate. directly from Springer, and in electronic form, or usual form, or maybe you may, you're Israeli. Why buy if you can get without payment? I can send you by PDF, and that's all. Just.
You download it for free from Springer?
Really?
Absolutely legally, in PDF form. I did it today.
Very strange business. I cannot understand it. But, okay, thank you.
And the last presentation that we have for today is from Dr. Ohad Gafni from Renewal Bio, Deep Biosolutions for Longevity. Please.
All right, so today I'm going to talk about how Renewal Bios is unleashing the true potential of pluripotent stem cells. So a little bit about Renewal Bio. So we've been founded about 1.5 years ago based on technology from Jacob Hanna's lab. So we got the IP from Weizmann, a combination of 3 breakthrough technologies that I'm going to expand during this talk. And we're mostly interested in cell replacement therapy, rejuvenation, aging, fertility. So what we have is a stemroid model that I'm going to elaborate that enables spontaneous and holistic differentiation of pluripotent stem cells and resulting with really authentic cells. So our mission is to provide patients and different technologies high quality cells for transplantations and also for rejuvenation of cellular and all kinds of different therapies. So there is unfulfilled promise of stem cell therapies for, and the audience here knows the different capabilities of pluripotent stem cells and what they can differentiate and what kinds of therapies they can provide. However, unfortunately, there are a lot of different, unfortunately, there are only a few approved therapies to date, despite having over 30 years of research and over 5,000, it's probably even higher today, of clinical trials. And the reason for that is in vitro differentiation is, and I think I'm kind of echoing what Yaky said before, that differentiation is very complex and it's very hard to do it in vitro. And I think that when we're doing in vitro differentiation, we're trying to push the fate of the cells into a different, like really certain direction, and we a lot of the times fail. And we're adding cytokines and morphogens and transcription factors, and eventually after a long period of time, this process is resulting with non-authentic cells, and these cells are not usable. And the problem is not with the pluripotent stem cells, because if we take the pluripotent stem cells, we put them into an embryo, we create a chimera and the cells differentiate very nicely. It doesn't matter if you're using mouse cells or even you're putting human cells in mouse embryos and human cells in pig embryos. The cells can differentiate, so the potential is there, it's just unleashed. So today I'm going to talk about how we're basically bridging this gap of availability of stem cells and the authenticity of stem cells. So we have a combination of 3 breakthrough technologies. One is basically culturing the cells in very unique naive media, growing embryos or embryo models outside of the uterus up to a specific point, and then producing stemroids, as we like to call them, but there are stem cell-derived embryo models. They're not embryos, they're stem cell-derived embryo models. So I'll start from the very beginning, sperm, oocyte, fertilization, create a zygote, 2-cell, 4-cell, so on and so forth, movula, blastocyst, but then, so we know very well how to grow embryos up to this point, whether different animals, humans. but there is a barrier starting after the post-implantation. So up to the pre-implantation, we know very well how to grow these, but after that, there's an issue. So we were able to grow blastocysts, derive them. Yamanaka in 2006 were able to take somatic cells and reprogram the somatic cells back into pluripotent stem cells, but they remain in prime state, and prime state is very limited and it's very committed into the specific fate, especially if you're doing reprogramming. Their epigenetic landscape kind of remains, and you need to reset that. And how do you do that? You kind of culture that with naive conditions. That's one of the breakthrough technologies from Jacob's Hannah's lab. The first was publication in 2013, but later on there were more developments. The second thing was, how do we cross this barrier? So this was resolved with the ex-uterine device. And then later on, something that was very interesting is, how do we make synthetic embryos or embryo models or stembroids, as I'll refer them from now on. How do we make those just from cells alone? So no oocyte, no sperm, no fertilization. And the interesting part here is we don't go through the pre-implantation stage. We go straight into post-implantation. And that's why these are not embryos. We can never put these stembroids into a surrogate mother because they won't implant. It's not possible, and of course we can't grow them to the whole gestation of humans. So this is a video of the capabilities of the ex-uterine device, and what happens here is basically we took embryos at mouse embryos at day 5.5 or 6.5, we grew them in this device right here. And what you can see here is the primitive string elongating and the somites created. And if you look further, you can see this is the point where we took them out of the uterus. So these were naturally conceived embryos, and they were put in this device, and they're of course growing, and you can see all of the elongation here of the primitive streak, and the somites would be here, and all of the formation of the heart and head and everything else. And if you remove the yolk sac, you can see better images. And this is how it looks. Formation, organogenesis, everything is happening, which was really exciting. So this is mouse, and this is naturally conceived embryo. So you're probably thinking, okay, this is good, but how are you going to use this? So the next thing that we did, there was actually a stage in between that we used mouse cells to create these stembroids. And actually, interestingly enough, the human cells, creating stembroids with human was a little bit easier because we didn't need exogenous factors to create the other cell types. So what we start with is naive pluripotent stem cells, but we induce them into two other, or maybe actually three other cell types. One is the hypoblast and the extraembryonic mesoderm, which would later form the yolk sac and umbilical cord, and the third one is the trophoblast, which would later form the placenta, and so on and so forth. And how does this work? We take these 3 or 4 cell types, we put them in a microwell of aggregation plate in a certain dilution, a certain number, and what you can see here is at day 0, they just look like cells. Day 1, they start to clump. Day 2, they start forming a ball. And by day 3, and you kind of bear with me and work your imagination, you can see that the epiblast is forming, the hypoblast is forming. Day 4, day 5, you can see the yolk sac or the primitive yolk sac and the chorion cavity. And day 6, day 7, day 8, they start and grow. And if you don't believe me, you can see here the staining. And what we've stained here is for, so you can see the DAPI and the white, that basically stain all of the nucleic acids. OCT4 is a specific stain for epiblast, SOC17 is for yolk sac, and CK7 is for trophoblast. As you can see here, day 4 of the protocol, at the lower panel, you can see a textbook images. Day 4 of the protocol is highly resemblant to 9 or 10 days post-fertilization, and you can see the epiblast here and the yolk sac forming, and also the yolk sac cavity starting to form here. And as we move to day 6, resembling 12 days post-fertilization, you can also start to see the current cavity forming and the primitive yolk sac expanding even more. And the cool part here is that we start to see lacunas. These are the structures that secrete the HCG. Later on day 8 of the protocol, we can see the 13, 14 resemblance and basically all of the different structures. So that was obviously very, very, very exciting. If we look further into this, we kind of took a home pregnancy test and we wanted to see if they actually secrete HCG, and they do. And I think that was very cool. But if we look further into them, you can see the really unique structures of our model, because we highly believe that the structure really governs the differentiation. And what we see here is the bilateral disc, which is very important for gastrulation, later organogenesis, and so on and so forth. But this structural development is important and unique in our differentiation method. And that's something that is very important to emphasize. So this, of course, made a lot of noise, and embryo models, and got the cover of Nature and Cell, and also some public press. And this is actually a non-published data that we got a few about a week ago. So I showed you day 13, 14 post-fertilization, and now we're talking about 19 post-fertilization. So we're moving on, and we can see the primitive streak and all that, and these structures are really interesting, because what you see here is this is the virtual human embryo atlas, and this atlas is basically from miscarriages. And what they did, they took the miscarriages and basically took them and made histology cuts and sections. So what you can see here is a actual embryo that was collected from miscarriage, and you can see here the connective stalk and the structure of the placenta here, the hypoblast here, and this is the actual embryo proper. This is the epiblast, and here you can see the amnion, and this is a stemroid, and you can see the disc here, the amnion here. This is the R placenta, and this is the yolk sac. So what we can do with this is, our vision is basically to take a skin biopsy from a patient and reprogram his somatic cells, whether it's blood cells or fibroblasts, and use non-integrative pluripotent reprogramming factors, the Yamanaka factors, grow them in primed, stabilize the conditions, then transition them into naive, stem them into the hypoblast and trophoblast induction that we're doing, to collect D3 cells and put them in the aggregation plate that we're doing, and then we're basically creating the stemroid by aggregation, mature them in our culture, ex utero culture device, up to the certain point that we need the certain cells for this patient, and then we can isolate the cells and put them back in the patient, and then you can have an autologous transplantation, no GVHD whatsoever. So that's our vision. That's what we're going to do. We're progressing, and we are isolating cells as we speak, endothelial cells, hematopoietic cells, because we can grow now. We're still characterizing this, but we can grow up to day 28 post-fertilization. Yeah, so we're really excited about what we're doing and what we're developing out of this, and hopefully by next year I could show you some in vivo functional data. And I think the really special thing about our system is it's not unilateral. What do I mean by that? So when in vitro differentiation, you get 1 cell type, 3 cell types most. With our platform, we're growing our stemroids, and we're getting more and more different cells that we didn't even think about. So we're growing, and we can see yolk sac cells, we can see endothelial cells. Now we're starting to see HSC cells, when we're actually seeing really specific endoderm cells that would later form hepatoblast and pancreatic cells. And later on, we're planning on getting oocytes and fertility cells, and of course, beta-secreting, insulin-secreting cells, beta cells. But we're really excited about the future. This is our team. With that, I would like to thank you, and thanks for staying so long.
In what type of extracellular matrix would you put the cell in this agri-well?
No extracellular matrix. No, but that's a really interesting question, because that part of our conversation from last night, we are trying different extracellular matrices to see if it improves the process.
Because we heard that the interstitial fluid is very important for the creation of the lumen.
Yep. And polarity and all that kind of stuff, yes, for sure. And
You get it just by putting them in a medium and agri-well.
yes, however, we do believe that the axis would be, so gastrulation is really governed by the axis. So that's what we need endometrial cells to see if they're working well or not. So yeah, that's an interesting question, and we're testing it.
Which day are you going to continue this differentiation process?
So
until which type of cells?
So the short answer is I don't know, but the long answer is, so we're not aiming to go past 140 days. But we know that we'll get to a certain point that now we're discovering more and more cells, so we can end up just cutting it at 100 days. But we are aiming to getting oocytes and stuff like that we know that usually you can get them at 70 days or later. But that's the thing about human development. So we know that we have an atlas of sections of H&E, but we know nothing about this process. And this is a really, really important process. And we know nothing about it since day 7, basically, since we implant the embryo up to day 100, we know very little. And we have very few references for different time points. So we have one embryo 16 to 19, we have one embryo 20 to 25, we have one embryo, and that's like the single cell resolution. But we know nothing about human embryo. We know nothing about human development at these time points. So I hope that we will also be able to kind of shed some light on these processes.
And can you comment a little bit about the ethical aspect of this?
Yeah, so we are we're head front on the ethical, and we're starting a committee about this, and the ISSCR is actually very active about embryo models today, because I think the overall notion is that these are not embryos. This is not something, and even in the UK, there is the 14-day rule, and for embryo models, it's not covering it. So you shouldn't consider it as embryos, because of course they cannot make a human. However, they can make really, really good cells from transplantation. And as Yaky mentioned before, I'm also a great believer in cells, and I think that's something that we should promote, because there are so many conditions that we can cure with really decent cells for cell therapy that I think the ethical ideas need to change in this notion, and they will change, and you already see the trend that it is changing.
So you avoid saying organ. You keep saying cells, because they will not get to the organization of an organ.
I think we can make cells and tissues. Organs, they need to develop even further, but there is a way to make organs, because there are a lot of companies that are decellularizing organs. So taking organs from a pig, decellularizing completely, and just staying with the scaffold, but they don't have a good source of repopulating these. We will be that source, and that's what we're going to do.
Thank you.
Yeah.
One more question. The FDA today is pushing quite significantly to reduce animal models and come with alternative. Are you considering also
Yes.
for screening method?
Yes, I love these questions, because that's something that we're also want to promote. We want to, and that's one thing. I worked a lot with pigs and mice, and I love that we don't use You know, even we don't need samples, basically. We just need fibroblasts or something like that. We don't need oocytes, we don't need sperms, and that's a platform that can definitely promote less animal use, and also a lot of the time the translation between mouse, rats, and humans doesn't work, you know, and that's solvable with this platform.
Thank you very much.
Thank you.
Thank you very much to all the people who remained to the last, who showed that the issue is really important. And we'll keep on strong and meet together tomorrow and the day after tomorrow and for many more years to come.